Go ahead.
Okay, so if people have questions while we're going through this, how's that process work for, excuse me. So today I have two basic topics that my slideshow will cover, and some of the content I'd really want to make sure I cover in the time that I have, and then there'll be some extra things that aren't part of the handouts that Michelle that are available to you, but are still worth thinking about, especially with this audience, which is professionally trained, formally trained in numerous aspects of crop management. And I think there's some things that we can talk about that go beyond what I might talk about to a group of growers, because as the people that are transferring this technology out to the clients, there are bigger questions that we have to consider that go beyond what we're telling the clients so that this technology is understandable and usable for them. So we'll get into some things going forward that are beyond just reading the tests, and some thoughts that I've been having as I do my research on what I'm looking at here. And it's at any point in the talk, please feel free to shout out a comment or ask Michelle to open up your mic, because the more we interact, the better it's going to be for understanding what's going on. So the first thing we'll talk about very briefly, and this is probably common knowledge to this group, but it's really something that's obviously at the core of a methodology that's analyzing the sap flow in a plant, and that is the relative mobility of minerals in a plant. And this is a topic that many scientists have spent many decades trying to understand, and the complexity alone of this particular topic is astounding. But the sap testing really offers us a way to peer into it in a way that we don't have with tissue tests. And I hope that that will be a little more clear as I continue and as I elaborate on it. But basically, the information on this chart is common knowledge, but it's a good thing to keep handy when you're working with a client or when you're interpreting a SAP test on the fly, especially if you're on the phone, because you can't always remember everything. I can't. Maybe somebody else can. Good for you, but I can't. But if you can remember this on the fly without having this chart, it can really help your mind go through the paces of trying to figure out what's going on sometimes. And this might be especially important in the case of, for instance, boron, which can be toxic, but is also essential for numerous aspects of the plant's physiology. And by knowing that it's not mobile, you can identify, say, a symptom at the meristem of a plant, or maybe something going on with flowering, or something else that comes to mind, and realize, okay, well, boron's not supposed to be mobile, but I know that it can move in phloem and xylem, which means that it can move through sap pretty well, depending on the circumstances, and that's an element that is kind of maligned and misunderstood, and it can be the root of a lot of problems. Sometimes toxicity related, sometimes phenologically related in terms of where the plant is vegetatively or reproductively. And that's one that I point that one out because it's one that we deal with quite often in Pennsylvania, trying to manage it so that it's sufficient but not toxic. Of course, nitrogen, potassium, and phosphorus are the big three, and they really come into importance because I don't know what your growers are doing to manage their nutrients, but excuse me, N, P, and K are a standard nutrient package for virtually all growers that are producing in the United States and elsewhere. They're a necessity for every crop. The other macronutrients and the micronutrients are also necessary, but in some cropping systems they don't get the attention that they get, say, in produce or in tree fruit. But having said all that, this testing method and the ability to gauge how nutrients are moving through sap eventually is going to give us tremendous insight into the entire plant soil system. Because as a consultant that I really admire in Nebraska told me once, a plant is really, all it's doing is using sunlight. It's harvesting sunlight to mine nutrients out of the soil. And if you think about it that way, that's pretty much what they do. And with that frame of reference, you can really look at everything else the plant is doing that we study and that we observe to produce a crop in terms of that simple metaphor. This chart or this graphic is going to be in your handouts, I believe. Is that right, Michelle? Well, I don't know if Michelle can hear me, but it's also part of the presentation that Gary is going to give, if I'm not mistaken. So we'll see that again. We might get into that more, but I'm not going to focus on it more today. Now we're going to start by looking at a test result for cherry tomatoes that came out of Lancaster County last year. When you get your sap analysis results back, you're going to see a page that looks like this. And just out of curiosity, has anybody actually run a sap analysis out of your program, Patrick? Not yet. Okay. Okay. So anyways, when you get your samples back or your test results back, this is what you'll see. Over here at the top of the page, there's information that you provide to the lab, and they will populate that part of the result with your ID information for your location, your grower, the field that's being sampled, and in that regard, it's no different than any other testing method. Over on the left-hand side, right here, you have the listing of nutrients and other parameters that are being measured in the sap. They include micronutrients, macronutrients, and then several measurements of nitrogen and different chemical properties of the sap, in addition to the solutes that are in the form of sucrose, glucose, fructose, and I believe that's it. They're mainly monosaccharides or disaccharides. They don't measure sorbitol or any of the sugar alcohols, and they don't record more complex carbohydrates like starch and some of those other stranger sugars that are found in certain crops only. But I think this sugar information is useful in some regard, and can be more useful as we learn more about these tests. So everything that's measured in a concentration is in a parts per million unit, which is, I like that because everything is comparable then. And on tissue tests, it's not a big deal, but I wish that a tissue test would give everything in one unit so that we weren't dealing with parts per million in percentage, because in my crude way of thinking, I like to see everything comparable one-to-one, so to speak. Now the EC at the third row down is electrical conductivity. That's measured in millisiemens per centimeter, and that's a measurement of the basically of the electrical of the voltage that can be carried by the ions, or the electrical charge that the ions are producing in the sap. It's a potential. And then the sugars are measured in percentage, which can be converted to parts per million if you desire, but I typically don't do that. And then this column are the values that are generated from the sap. So that's what I call the metric, or the value of the metric. And then on the right-hand side of the result, you're going to see a column on the left, which is what Nova Crop Controls considers low, and in the lower end of that range, deficiency. And the middle column represents what they consider sufficient and desirable for the different metrics. And then the upper range on the far right is what is considered excessive or high. And that's not necessarily bad. That's why I have it yellow. You know, it's like a yellow light, caution. Maybe there's something going on here that's good or bad. Let's think about it. Now, we're going to focus on cations on this particular slide. So the major cations obviously are potassium, calcium, and magnesium, although sodium is also represented on these results. And in Pennsylvania, sodium is typically not a player. It could be in other parts of the country, and of course that's has an effect on cation balance, because ions of like charge they can antagonize each other for positions in the sap, in the plant, on the receptors of enzymes, or in other areas of the plant physiology. And in fact, there are some receptors that can take, say, potassium or sodium, and in some plants they actually do better if the sodium's out competing the potassium. So sodium can be important in some cases, but generally we focus on potassium, calcium, and magnesium, and the potassium to calcium ratio, which is a crude measurement of, in some cases, what kind of fruit quality you can expect, especially in apples, depending on the variety. Now, on any sap test where there are target values, that's what these green bars are. They're called target values. They're based on what Nova Crop Control has determined are the ranges where these nutrients should be in sap to have crops that are functioning at their optimum. And they've based these target value measurements on numerous observations over many years, and they're very, very confident in them, and they don't release them unless they are confident that it's a pretty precise representation of what the plants need and the importance of that nutrient in the plant. On the target value bars, the top bar is numbered 1, and that represents the value of the amount of that particular nutrient in the young sap, and it's always going to be light green. And the bottom bar is a dark green bar numbered 2, which represents the mineral concentration in the old sap. And I won't get too much into sampling at this stage of the slideshow, but there are protocols for every crop and every type of plant so that the best possible old and young sap samples can be collected. So generally, when we're talking about these cations, we'd like to see that the potassium and magnesium are greater in the old sap, because they're very mobile in the plant. And in a nutshell, if the plant is at its optimum, or it's functioning properly, it can really self-regulate these nutrients if they're available. And if it's sensing a deficiency in its younger foliage or in its younger sink, whether that be buds, meristematic tissue, or fruit, or leaves, plants are very good at redirecting nutrients from old areas to new areas. And the highly mobile nutrients have this characteristic, and magnesium and potassium are in that category among the cations. And this is a tomato test result. I didn't emphasize that, but these are cherry tomatoes from the owner of Keystone Bioag. He did a very nice big sample set last year, and in tomatoes, any fruiting crop, the potassium to calcium ratio is a key indicator of what fruit quality you can expect. And generally it's desirable to have it around 1, because if there's too much calcium, then potassium can be excluded, and that causes a series of conditions in the fruit that are undesirable for fruit finish and quality, and the fruit won't be as aesthetically pleasing or as flavorful. But if the potassium is too high and that ratio is falling on that side of the relationship, there could be problems with the integrity of the cells and the shape of the fruit. And in tomatoes, there's a condition called blossom end rot, which is a result of imbalances between these cations that actually open the fruit up at the calyx. And at the bottom part of the tomato, where the flower used to be, there's a little black spot on the bottom of the tomato. That's called the calyx. And if the potassium-calcium ratio is too high in favor of potassium, that can open up and allow in disease organisms and just cause a black, corky spot that it's unsellable, really. So in tomatoes in particular, that's something that we examine, and we do in tree fruit for very similar reasons, although the imbalance manifests differently in apples. So does anybody have any questions up to this point on the cation part of the test? I will say though that before we leave this slide, what we have going on here is actually a pretty good situation, as you can see. Sam Zook is the fellow who generated this test. He does a pretty nice job of managing his crops. Doesn't use pesticides, uses very little nitrogen input, and his potassium levels are very good. Here, as you can see, the younger sap has less than the older sap, which means that there's a steady supply of potassium moving through that sap, and it's being replaced, and that there's not a deficiency imminent. The magnesium is the same, and typically with magnesium we don't see a huge difference between the young and the old sap levels like you see with potassium and calcium. For some reason, magnesium tends to be within a few hundred parts per million in the old and young sap. I haven't figured out why that is, but it makes managing magnesium rather easy for us in Pennsylvania, because we don't have very good standalone magnesium products that we can use if we're deficient in that, because most of the products that are being used in the systems where I work have something else in them that you don't necessarily want your crops exposed to at that time of the season. And then with regard to the potassium calcium ratio, it's at this stage of the game it's very close to where we want it. And I'm not sure exactly when this sample was taken, but it was probably in May or June when the plants didn't have a heavy fruit load. So I would expect that. And we'll talk about some other trends in cations later in this part of the talk, so I'm going to move on, unless someone wants to interject something. Now this is Sam's entire data set for his Jolly Elves from the beginning of June until the beginning of October. This is an ideal data set, and since Sam, he does a lot of his own investigations on his farm using these methods so that he can understand what they're telling him and how to manage his crops. You probably won't see this from too many growers, but it's a nice illustration of why a bigger data set is really good for this method. And as you can see here, the target values that Nova Crop Control has established for potassium on tomatoes is roughly 3,000 to 6,000 parts per million. So if your potassium levels for this crop are within that range, that's a really good sign that your potassium management is right where you want it to be for this crop. And it's clear here that the levels of potassium in the young sap were sufficient. They were not getting low to the point where the old sap was being tapped, except there at the middle of July. And Sam does add a very small rate of potassium to his tomatoes weekly, but generally he didn't overdose them. And it's very evident that there was plenty of potassium in those plants. And you can never really be 100% sure why that's happening in a system, any cropping system, because it's so complex and you can't measure everything. But I attribute a lot of that to his soil management, because he doesn't use any pesticides, and he uses a lot of highly mature humus compost and very good soil amendments that feed soil microbes and maintain a very, very good soil microbial population, which is doing a big job for him in making nutrients available. And potassium's not the first thing you think of with nutrient cycling in soil, but there's no question that there's a large pool of potassium, and it's available to his plants, and it's a testament to his management, which involves good irrigation and very careful stewardship of the livestock under the soil, so to speak. This is a picture of the tomatoes at the time they were being harvested, and they are in really good shape for this stage of the season. And there are lots of tomatoes. It's a variety that really bears heavily, and these are good quality tomatoes. He was filling buckets, and his nutrient inputs were very minimal, and he did a lot of sap testing to refine his micronutrient input and his overall management. He's a very successful tomato grower, and that's saying a lot in southern Pennsylvania, where there's a tomato grower underneath every rock. Now, if we look at his calcium, we see some patterns there that are not unexpected, and this is very common in Pennsylvania with all the sap testing that we see, where the old leaves have an overabundance of calcium, and the young leaves are never deficient, and they're never excessive. They're always in that zone where you want them to be, and this shouldn't surprise anyone because calcium's not mobile in phloem. So there's only one way that the plant is picking it up, that's through the xylem. And again, when Sam's watering schedule and his inputs were helping his soil make it available to his roots, and he managed to have a very good balance between his calcium and potassium. And I say balance, I don't always like to use that word because it's like, what's balance? You know, how do you determine that? But when you have good fruit quality and you can see that your management is keeping these nutrients at levels where the desired ranges would like to see them, then you're achieving a balance in the, you know, in the very loose sense. So most calcium will probably, most calcium profiles on SAP tests will look like this over time. Although it definitely varies by soil, and in Pennsylvania we have very calcareous soils with limestone faces. So that is a big part of why we see this. In other areas, obviously, it'll be different. Now, this is Sam has a story about this high tunnel, and on the right-hand side of the tunnel, there are some plants that look like they're in really bad shape, and on the left-hand side, they're not much better, but they're not worse. And Sam likes to joke that the grower told him that when this picture was taken, those tomato plants were 3 times better than they were the year before. And Sam puts these slides in here to illustrate how important soil management is. And in the slide before this, he had actually recommended this guy remove his soil and just put new soil in because his soil was so, I don't know what had happened to it, but he wasn't getting any production. And a SAP test, I don't remember if they did SAP testing in here, but you would have had a very different profile than what Sam had. Now, this is not the same grower, but this is one of the top tomato growers in Lancaster County. Routinely will get over $100 a box at tomato season when everybody and his brother is bringing tomatoes to the auction. And this is a grower who uses SAP testing on the regular basis and will not go back to tissue sampling. So it's really made an impact with the produce growers in southern PA. And it's starting to do the same across the country with people who are working with it. Pardon me, I had to have a drink of water. How are we doing for time, Michelle?
We're at 1 o'clock now, so if you want to continue for another 15 minutes, 25 minutes, we'll reserve some time for questions and then let Patrick and his group take a half an hour for break for lunch.
Okay, I won't be able to get through everything in 15 minutes, so I'll
Well, let's do 25 then, 25 to half an hour.
I'll try speeding it up. How's that?
Okay, very good.
That's what I'll do. Okay. So now we'll look at some nitrogen values, and obviously nitrogen is, next to carbon, is the most important nutrient that plants will consume for numerous reasons. And we have 3 different measurements of nitrogen, and one of them, okay, somebody just said we don't need to eat. Am I reading that right? Oh, it says we don't need much time, but we still need to eat. Roger that. Okay. So anyways, there are actually 4 metrics for nitrogen, but 3 different nitrogen values. There's ammonium, nitrate, nitrogen in nitrate, and total nitrogen. Now, depending on the consultant you talk to, he or she will focus on N in nitrate, but the more I study these tests and learn about the interactions of the nutrients, I'm starting to look more at the nitrate levels themselves. And the N in nitrate it's not actually a measured value, it's a calculated value based on the nitrate number. So I'm tending to focus more on nitrate values because when you look at the scientific literature, they talk about nitrate nitrogen as NO3, as that ion. So I think it's important for me as a consultant to put what I'm talking about in the nitrogen in that form, in that context, and with these tests especially, because when scientists study nitrogen in sap or in cells, they talk about nitrate nitrogen. So at any rate, generally what the consultants will say that are using this testing is they want ammonium levels below 100 parts per million and nitrate in the low or optimal range, and obviously total nitrogen in the optimal range. Now, all of these forms of nitrogen are, well, nitrate especially, are pretty mobile. That can really be moved pretty quickly through the xylem, and there's evidence through phloem. So there's generally generally nitrogen is in good supply in all cropping systems that I see, and it's usually sufficient in the old versus young sap in the sense that the young sap is not showing a high amount of nitrate, indicating it's being pulled out of the older parts of the plant. Now, in this particular case, the nitrogen is rather low, and that's desirable because if it's low, that means generally it's being processed, it's being reduced into ammonium, which is then being burned up through the enzymatic machinery to make amino acids. And the amino acid complement isn't measured directly, but you can gauge the performance of the plant by the total nitrogen and how the two inorganic nitrogen forms measure to the total nitrogen. Now, these plants are young. This was in June, before they really started to put on fruit. So it doesn't surprise me that the total nitrogen is largely made up of the inorganic forms. As the season draws on, you'll start to see that change, and the ammonium levels will fluctuate. But generally, what I see is that total nitrogen tends to be in the range where you want it, and the inorganic nitrogen is a small percentage of that total nitrogen. And that's desirable because that indicates that there's a good complement of amino acids and polypeptides mainly, and some protein, because in plant sap, the nitrogen that's not in an organic form is being transported largely in the form of amino acids. So I'm putting check marks here to show that yes, these are at levels that are desirable and optimal, and that the nitrogen management at this point in the season was very good, and that we know that there's sufficient nitrogen in that sap moving around to the places where it needs to go, probably at this stage of the game into the meristem, and that you would not expect to see deficiencies in the plants a week from now. In a nutshell, sulfur is not a mobile nutrient. So if it's deficient in young leaves, it will not be resupplied by the old leaves. And what this is indicating is that there's a supply of sulfate ions providing sulfur to the sap. And these levels don't change very much over season, which is indicative of a constant supply of this in the sap that's moving where the plant needs it. But generally, I should stop saying generally, I'm sounding like a broken record, but usually what we see is more sulfur in the old sap, although that can vary by crop. But you might expect that because it's not mobile, and if the older leaves have the complement of sulfur that they need, it might start to accumulate in that older sap rather than being transported and consumed. And with micronutrients, you see different patterns, even though they're like sulfur in the fact that they're not necessarily mobile, but you do see some interesting things showing up with their presence in the sap. I put a red X beside this because that level in the young tissue and young sap is lower than Nova Crop Control recommends. But how you address it is difficult because there's not a very good product that will supply sulfur that we're aware of. Now, in terms of phosphorus, that is a highly mobile nutrient. So what we usually see in our soil, our plants, is a lower amount in the old leaves because it's being consumed. It moves through the sap into the younger uh, parts of the plant, and they are constantly drawing it off because it's not very available through the soil. But the goal in our area is greater than 200 parts per million in old and young sap. And one of the goals Sam has this year is to boost that as much as possible through a variety of methods. Again, it's not optimal. It's often what we see, and it's something that we are really trying to address and using the sap testing to do that. And here's another graph. Now you're familiar with these. At this later part of the season, the values, the phosphorus levels approached each other a little better than they had been earlier. Is having them 100 parts per million apart, even if it's a deficiency in the younger or the older sap, desirable? We don't know. We have to continue looking at these to find out if we can determine a relationship like that. Now, micronutrients are, I like to call them a gray area because there are so many different ways they can interact and be affected by different nutrients and other things going on in the plant. But in our area, it's validating the rainbow-colored graph that shows that at lower pHs, micronutrients are more available, with the exception of molybdenum. That's often the case with our samples and our soils in Pennsylvania. But the recommendation from Nova Crop Control is to have boron at less than 12 parts per million to prevent toxicity, although it can be higher without being necessarily toxic. And to keep manganese around 15 parts per million, which isn't very hard to do if you have good standalone manganese products. In this particular example, the manganese is low in the young sap and approaching low in the old sap. And if Sam wouldn't address this, there's a good chance that the next time he sampled, this manganese would be low in both as it's being consumed by the plant, because there wouldn't be anything to replace what's moving out of the sap into the cells. And so I'm sure that Sam addressed this, although I don't remember exactly what he did. Now here are the micronutrient target values as established by Nova Crop Control for each of the major micronutrients. And this is in your handout, so I won't elaborate any more on this. And now we're going to look at a quick profile of the cations over the entire course of the sap testing. And this is also part of your handout, but on the next slide I'll actually summarize some of the trends that were in there. And if you look at this, just study it for a minute, you'll see some patterns in the concentrations of nutrients in this sap, which is indicative of his management and his soil quality. But there are a lot of similarities here across the time points. And those are that the magnesium doesn't really, it's in a narrow range, about 300 parts per million for the entire season. Calcium is always higher in the old sap because it's accumulating in the leaves and it's not being transported out to the the fruit and to the flowers and to the other parts of the plant that could use it, which makes it problematic sometimes. And the potassium again was managed so that it was higher in the old sap the entire season, which provided a reservoir and an excess in the old sap, if you will, that was always ready to replenish what was being consumed by the fruit and by the meristem and by the leaves. If there was a challenge by a mite or disease, potassium is implicated with plant defense, so it's good to have that potassium ratio between the old and young sap to where it was. Now, Michelle, I can go one of two directions. I can talk about the bell peppers, or we can talk about the broccoli. It depends on how much time we have. Tell me how much time I have. Is anybody there? Hello. Okay, roger that. So another crop where there are established target values, which are very reliable, is bell peppers. And this was, we're going to see a series of charts from a pepper grower in near Sam, who is a top-notch grower, and he had excellent management, which this series of charts will show us. It will also show us some very distinctive differences between peppers and tomatoes. First of all, you'll notice that the potassium levels are, if you would look at this and you didn't know, you would immediately say that, well, there's something's wrong. There's no potassium or no calcium, and it's much lower in the old sap, which is something we don't expect to see. And that's not unusual for peppers. In fact, we're of the mind that, and these test results confirm that peppers are heavy potassium consumers, and they are always going to have more potassium than calcium in their sap and in their fruit everywhere in the plant. The potassium to calcium ratios will always be sky high, and you don't often see this in a crop. It's very rare to see it happen naturally without excessive input, but in bell peppers it's the normal. And if you'll notice on this test, even though the potassium and calcium ratio is so high in favor of potassium, you'll notice that you would think, well, calcium's insufficient. I mean, there's not enough there at 38 and 19 parts per million, but you'll notice that the Nova crop control target value actually is a lot lower for calcium. So it's actually not terribly out of balance in terms of where it should be for sufficiency, but because the potassium is so competitive and so dominant in this system, it's excluding calcium and creating a really situation where there's a pretty big difference between the old and the young sap in where the calcium is actually showing up in the sap. So I haven't quite figured out why this is going on in peppers. It's not something that I have thought much about yet, but it's something we need to consider because blossom end rot is can be a serious problem with pepper growers. But you'll notice the magnesium is not too much different from the what we saw in the tomato data set. Now I'll take this minute to talk about ammonium. As I mentioned before, Nova Crop Control stresses low ammonium levels in plants, and ammonium is an important molecule, and it has a lot of different functions in plants, and it can be generated by plants in a number of different ways in addition to being taken up through the soil. So as I read more about it, my opinion is that we don't need to be as focused on ammonium as a detriment to a plant as you'll hear. I think that there's reason to be concerned if it gets high enough to be toxic, but I haven't seen that. And if you read about ammonium processing in plants, you'll realize that they very quickly can quell an ammonium excess, especially if they're making it through proteolysis or through photorespiration or some other process that generates it naturally. It's a natural component of plant physiology that shouldn't be treated like some sort of bad guy that's going to create all kinds of problems for you. I don't believe that at all. But having said that, it's important to watch the levels of ammonium and manage the plants in a way that can optimize them to use it to make amino acids so that it doesn't get to toxic levels. The reason it's high, though, in this sample is not because this plant is sick or because it's creating excessive ammonium. It's because the sample got held up at customs in the Netherlands, and as the proteins were breaking down, they made ammonium that showed up in the test result. And since the leaves weren't active anymore, the enzymatic machinery to process the ammonium wasn't active, and it built up. You don't like to see it on a plant. If you see this for several samples in a row where there hasn't been a holdup, there's going to be an issue, but it wasn't an issue in this particular case. And here's an example of why. The following tests, we just didn't see a buildup. It was actually very low in subsequent testing once the samples were being processed in a timely manner. As you can see, it was well below 500 parts per million, and at the very end of the season was even below 2. Now, if you look at this nutrient profile, I'm a little short on time, so I can't cover everything I'd like to. There's some very interesting things going on, but you will notice here that, again, the ammonium was high according to the chart, but it was not high according to what's physiologically acceptable. You'll see, of course, the potassium and calcium ratios were very high in favor of potassium, but the calcium levels had increased, and I believe this is because the grower was applying calcium in an effort to mitigate that high ratio, but it didn't really. And there's probably no need to be excessive with calcium, but you certainly want to manage your plants so that sap has calcium in that sufficiency range, so that you know that there's enough calcium available in that xylem to supply the plant with the basic needs that it has. On this particular test, a series of shots, you'll see that the micronutrients seem to be pretty deficient with the exception of iron, although the iron levels did, for some reason, they were higher on the 29th of June than they were 2 weeks later. And iron is a funny nutrient because it can, depending on what other micronutrients are doing, iron can be in short supply or could be excessive. It gets carried around by organic acids and amino acids in the sap, so that it's being shunted around, carried, and as it's being consumed, it's, in my mind, I'm not sure if a low iron level means that the plant doesn't need it because it's already got what it needs in its cells, or that it's not taking it up. And that's a huge question that all of us working with this methodology need to be keeping in mind as we use it with our clients. It's not necessarily something we have to discuss with them because it might be a little academic for them, but privately, when we have meetings like this, we should be thinking about what the nutrients mean in terms of being part of a system that's moving through the plant. Because if you don't look at it that way, this quickly becomes a $75 tissue test. We can take a break anytime. Oh, Michelle's sending me a private message. I just blew that one up. Sorry. Michelle's indicated that we could take a break. I'm fine with that. And if I have to stop here, I can. But I can continue afterwards if we want to do that.
Patrick, how's your group?
I think we're doing fine. It's 11:15 here, so.
Okay.
We have 45 minutes, right?
Would it be appropriate for your group to take a break, or would you prefer that we just keep going?
No, I think we're fine. Keep going.
Okay, wonderful. Go ahead, Nick.
All right. So getting back to the point I was making before I interrupted myself, I'm starting to think of the sap testing as a way to look at a dynamic part of the plant that up to this point hasn't been measurable before. And I'm really trying to understand more about how nutrients get into plants and what happens to them after they're in the xylem and phloem. How do they get there? What's influencing that process? And where they're going once they get into the sap. And that's a huge task to undertake because every crop has this nutrient profile and they're all going to be different. But that's where the real power is in this method. And that's where, if we can understand that and articulate it in a way that a grower can use that information to make a decision that improves his outcome, it's a grand slam. And that's my goal. And it starts with one step, but I've taken that one step, and it's actually very fascinating when you start to study the resources that are available from 60 years of plant science in the modern era to understand this. And you're going to see things on these results that might raise questions in your mind, and I don't cover everything because we don't have time. And as I said, it becomes academic after a while. But as you look at these, you'll start to see things on the tests in the context of what we've already talked about that start to make sense. And a green plant is a green plant, whether it's got peppers hanging off of it or tomatoes or apples or whether it's broccoli. There are certain things that are going to be very similar across cropping groups and plant families. And they're very easily identifiable if you look at enough of these tests. For instance, on this one, you can see on this that the nitrate levels they would be considered, maybe 700 parts per million actually that wouldn't be. See, I'm getting ahead of myself. But if you look at the nitrate levels, they're very low. And in Pennsylvania, what we often see once the plant is mature and is actually reproductive is that they are often less than 20, sometimes less than 5 in the N and nitrate value. But what I typically see is that total nitrogen is usually somewhere around the sufficiency range or just below it in terms of how much of that total nitrogen in the form of amino acids, polypeptides, and proteins is in the sap that's being moved to where it's needed. In this particular example, it's higher in the young sap, which doesn't surprise me because these are pepper plants that are in their reproductive stage right now. So the peppers are consuming quite a bit of nitrogen, and there's a lot of it moving up there. And getting back to what I said about the sap as a transport system, we also should really be thinking about sources and sinks, and those are two terms that have been adopted by the plant physiology community to describe where things are made as opposed to where they go. And if you know that at a particular stage in a plant's growth that it's reproductive, but it's not a fruit that's maturing, say, like a pepper, you know that if you see this pattern in the sap with total nitrogen, you know that that plant is exporting things from where they're being made through the sap into the fruit, and that's why you see that higher number in the young sap, because that young sap is constantly providing nutrients in the form of amino acids and polypeptides to that fruit. So in this case, you can understand why it's higher in the young sap. And there's no question about why it's higher. With some of the micronutrients, which are kind of nebulous in with some of the micronutrients, they're needed for enzymes in particular. So like I said before, when a micronutrient level is low in sap, does that mean that the plant can't use it at that stage, or does that mean it's not there? You don't have that question with nitrogen. I can pretty much guarantee that nitrogen is being consumed from leaves, from photorespiration, from some other place, or being drawn up from the soil, and it's being processed and sent to meristems, to flowers, to seeds, and to fruit. Does that make sense? That's a good question. Can everybody see that question?
I can. Gary, can you see it?
Yes.
Should I type a reply in there?
Just go ahead and answer the question verbally. That'd be great.
Okay. There's a question about whether there are guides to understand when these source sink shifts occur, and I don't know. And I don't know if there's a comprehensive source of that particular aspect of plant development in the context of nutrients.
If I could
I'd like
if I could speak to that briefly, Patrick, what we're anticipating or what we're establishing for your group specifically for potatoes is direct dialogue with the Nova Crop Lab, and they do have. I can't guarantee that it is a printout or a concise guide, but it is the access to the consultants who have developed the technology to be able to detail for you the trends that they have observed and that they have with their 10 years of data collection.
and
So
it's
basically,
yeah, and with potatoes, that's especially important with potatoes because the first tuber is going to dictate what the like I don't know much about potato production, but from what I know, that first tuber's um, initiation and development is critical for the rest of the yield. And that tuber is a huge sink. It's a huge sink for sugars and micronutrients. And I know I read somewhere that if copper is excessive in a potato plant, it'll build up in the tuber. So I don't know if that's good or bad from the perspective of potatoes that are going to be made into chips or they're going to be like harvested for fresh consumption. But I can't. It would surprise me if we can't figure out how to predict that or see that happening in real time with these tests. I'm not saying we can because I don't know, but I would be very surprised if we couldn't figure it out somehow with after doing this for many growers in different fields over several seasons. That again is like one of the goals in my mind for this technology. I think that's where we can go with it.
Hey Nick, this is Patrick.
Yeah.
So I think something for dialogue with Nova Crop and for the other people who are thinking about potatoes or whatever crop it may be, having something like a growth curve and where when you're going along a growth curve, whether you're at the early vegetative or mid vegetative or flowering or fruit set phase, just to know what are the shifts that we should be watching for, which direction should these vectors be heading? Is it up or down at these particular moments? That way, as we get into the season, we have a sort of a guide as to say, are we pointed in the right direction at this moment in time?
Yeah, that's a good point. And I've seen those for tomatoes and corn and cotton here in the States. And there might be something out there like that for potatoes because it's such an important staple crop, and the one thing that I find frustrating though with getting into the SAP testing though is that there hasn't been a lot of research done on the questions we're asking in terms of like using SAP to figure it out. So basically a question like that, Patrick, has got to be answered from the ground up if we can't find that someplace. And asking Nova Crop Control is a good place to start because they've done the research. And it's going to take a while to get questions like that answered across the board for the different crops we work in, but at least you're asking them. And that's what's important because if nobody asks it, we can't even begin to tackle it. So I wrote it down as something I'll try to find out more about. Now, I'm going to point out a few things here. I know I'm running low on time, but I'm going to get through the stuff I really wanted to. But let me just change gears for a minute and talk a little bit about total sugars, pH, and electrical conductivity. I won't get too much into this for just to save time, but our experience is that pH doesn't change much in the sap. I think that's a really powerful statement on the plant's ability to regulate its internal pH by a variety of methods. Now, sugar is a different story. Sugars can vary by the time of day. They can vary by the level of nitrogen, by the level of potassium. In cucurbits, I know Michelle's sound like a broken record because she's heard me say this more than once in the last week. In cucurbits, their predominant sugar is a sugar alcohol, which this test doesn't even measure. So cucurbits always have a low sugar level, which looks like they're not photosynthesizing properly, but they really are. We can't look at it with this test. The electrical conductivity as a practical metric has very little significance with these tests other than when it's above 15, just arbitrarily. That's the number they gave me at the lab. If it's above 15, there's probably something going on with the cation and anion ratios that needs to be examined. And in this case, potassium is really probably pushing that, and chlorine is too. Chlorine is actually a nutrient that plants need. It has a role in photosynthesis, and it's also important for osmoregulation. So with these pepper plants, I'm not sure why the chlorine is so high in the sap. It's obviously there's a source of it someplace, and it's being put into the plant. Maybe his water has chlorine in it. I don't know. It's possible that the plant is scavenging it from. Maybe from that. That's not. I'm just kind of thinking out loud. But let's just say hypothetically that the plant is trying to create a better cation to anion ratio for its development. It's not, in my mind, from what I know, it's not inconceivable that you would see some of these ions at higher levels in an extreme case like this where potassium dominates. Oh yeah, go ahead. Oh, that's what it says. That's right. Unmuted. Oh, that's what it says. That's right.
Hi, Gary. I unmuted you if you had something to add to what Nick's talking about.
We moved on since I might address it later on. As far as the reproductive vegetative growth shifts that occur in the potatoes, we're working with some seed potato farmers, and we had a little discussion about that and what impact that has, and that plants actually shift between vegetative and reproductive multiple times throughout their season, especially those that continue to bear, like potatoes that put on 2 or maybe even 3 sets of tubers, or tomatoes that continually blossom and fruit throughout the season. So those are some things we can discuss later too.
Yeah, that's a good point. Because sugar and nitrogen, from my understanding, they dominate a lot of what happens in a plant because for obvious reasons. And it's my opinion that as we learn more about these tests and learn more about what they're telling us about the plants, that we'll be able to piece together what science knows about sugar in plants, and if we tease these apart enough, we'll start to see patterns that will shed light on areas where we can influence the plants, like Gary's talking about, like these switches that go on and off between vegetative and reproductive stages. Okay, so
Nick, does that answer Patrick's question about the non-variation of sugar across the sample dates?
Nick, this is Patrick. I was just wondering, while you were talking, you mentioned that sugar can go up and down, but it's printed on the slide that it doesn't vary much across sample dates. Just want a little bit of clarification.
Did I say that? One of my observation is that it generally doesn't. If the plants are sampled at the same time of day by that grower, they're not going to be very different. But the plants, now from what I understand about plant physiology, they will, sugars are an important part of how they, operate their machinery, so to speak. And I know that if you sample plants later in the day, there's going to be more sugar in the sap because by the end of the light cycle in the late afternoon, the plant has been able to transport sugar up through its sap. So if you don't sample your plants at the same time of day every time you do it, you'll see that changes that might be false positives, so to speak. But Patrick, that's a good question. I don't.
Yeah, we'll just have to watch and see. I know from doing Brix testing in field, we find variation, and not just based on time of day, but also on soil moisture conditions. So that's something we're going to need to sort out on our part. Is we won't always be able to get into the field to sample it when the moisture is exactly the same as the last time we were there to sample it. So,
yeah, that's a good point. That's a good point. And I guess the take-home message on the sugars is that, in my mind, they're underutilized on these tests. And while, like exactly what you said, moisture, the soil moisture, that's why I'm starting to think more on my own that the whole plant system can be represented. The whole plant system can be represented in these metrics. So as I'm just emphasizing here that the chlorine is high, and it's high relative to the sulfur and the phosphorus, and that's probably part of the reason that the EC has been driven up in the old sap and the new sap. So the next question is, well, what does the grower do about it? And that depends on the situation and the means at his disposal to address a problem like that. So now we go another 4 weeks into the future here, and you see that there are some very dramatic differences in the nutrient profiles here. Gary's unmuting.
Go ahead, Gary.
Fire away, Gary.
Unmuted. There you go.
Fire away, Gary.
I was just going to say that I've got a sample on potatoes that we have 4 data points through the season on in sap, and he went from a 0.2 to 0.4 range on sugars to the end where we got clear up to 2 on some of the new leaves and over 0.6 on the old. So sugars will move during the season depending on what's going on and depending on the plant and the health of the plant, I think. But we can influence that with nutrition as well.
And that gets to a bigger question that I really have in my own mind going forward with the SAP testing in regard to sugar and plant health, if you will, which is a term that can be defined several ways depending on who you talk to. But in our sense, it's one of the things about plant health that I have in my mind is how can it meet the challenge of an insect or disease on its own without fungicidal or insecticidal input? And that's a question you could ask about any plant. That's an interesting question in itself. And sugar or carbohydrate to nitrogen ratios are key in the relationships between plants and insects or other arthropods. That's been demonstrated in the literature time and time again over the last 50 years or so. So hopefully there will be a point when these tests will help us figure out what that means in terms of potential insect attack, and with the potatoes, perhaps managing that psyllid that spreads the zebra chip bacterium. I don't know. It's if the research isn't out there, we're not going to be able to tell from the sap test. But at least we have something that we could piece together with good research if somebody takes it down that road, looking at how what's in that xylem and phloem is influencing feeding and transmission of those diseases by the psyllids. We're not there yet, and it'll be a long time before we get there, but I feel like this is a tool that could help us get there. And I don't think we'll have time today to get into it, but I have slides that highlight that a little more later. I just want to make sure I get through what I'm committed to by Michelle, and I'm going to do that now. But what really is impressive about this particular data set is that the micronutrient levels have really improved, and that's because he was putting micronutrients through his drip lines and probably spraying them too. And you can see the huge impact that has. If we go back and look at this one before, you see that they're getting higher. The sap is taking them in. They're being relocated through the plant. But if you go to the first test that we had, see that, look how low they are. So the grower had access to standalone micronutrients, and it's obvious that he was availing himself of those. And there are numerous interactions that micronutrients have with each other in the plant and in sap, and unfortunately that's a whole hour in itself to go over that, but suffice it to say that if you could have one micronutrient that's very high that could suppress others. There's a relationship between phosphorus and zinc that's very important. If there's an excessive phosphorus in the sap, that can indicate that zinc might not be as available to the plant unless it's being added. Now, in this particular example, when his phosphorus levels were improved because of his soil management and his input, his zinc levels were still solid in the sap because he was supplying zinc to the plants. And zinc is very crucial in meristematic tissue. In fact, there could be a higher percentage of it there than other places in the plant. And adequate zinc, even at its lowest level of adequacy, it will really boost photosynthesis, as will many of these micronutrients. So when the zinc levels are in the sap as the amendment, you see that they're higher in the young sap, which shouldn't be surprising because, like the nitrogen, they're being sent where there's the greatest need for them. Copper is very similar. It's mobile to some extent, and if I'm not mistaken, copper will be relocated into fruit as fruit matures. Not in great quantities, but if that's true, if I'm remembering correctly, that would make total sense with the copper being high in this young sap, wouldn't it? Because that is up there near the peppers. It's receiving solutes from the old sap and shunting them into the reproductive structures. Something else came to mind I wanted to mention, but it's eluding me now. Oh, I know what it was. If you look at, and here's I'm going to go back and man up and say I was wrong about the sugars, because as the micronutrient levels in the sap improved and the cells were getting what they needed, and I think some of this high micronutrient content is reflective of the cells not necessarily needing all these micronutrients, and they're just kind of circulating in the sap, although that's speculation. You notice that the sugar levels jumped. And as Gary had mentioned in the potatoes, he said that he sees young sap sugar exceeding old sap sugar in the reproductive phases, and that's what... Oh, I see there's a question. Sorry, I was running my mouth and I didn't see it. The question is, on micronutrient additions, how did the farmer decide how much of a particular product to add? Well, basically it was label rate. It was guesswork because this is the first year that Sam had micronutrient products that he could sell these guys. This is one of Sam's clients. So we didn't know what to expect. Had no idea how the plants would respond and how the sap test would show. But as you can see, even a pint per acre through the drip had a dramatic effect. So this coming season, my guess is that Sam is going to try to fine-tune these rates a little bit with his guys, and with some of the more experienced growers, see what the minimum is that they can add. That'd be my guess, because it doesn't take much. There is kind of a good news, bad news situation though with the micronutrients related to insects, and I've got to talk about bugs at least once before we quit, in addition to the psyllids. Micronutrients are essential for enzymatic activity, as we all know. And there's an enzyme called rubisco, which is critical for the Calvin cycle. And it has requirements for micronutrients. And the good news, bad news situation is that to optimize the plant's photosynthetic capability, there has to be a good complement of rubisco in the cells. But rubisco is a protein. It's probably the most abundant protein on Earth because it's in plants. And when insects feed, especially like tobacco hornworms, for instance, that's where they're getting their protein, because it's been shown in studies that rubisco, when caterpillars, Manduca sexta caterpillars have been fed rubisco and leaf tissue, that it doesn't show up in their frass, which means they're digesting it. So it's a good news, bad news situation because you've got to have it there, but you know it's something that the bugs are going to seek out. And if you look at insect feeding biology, you see that across the board. What's good for the plant is good for them too. And so, as I said before, with the carbon to nitrogen ratio, that's a key component of understanding how a plant is attractive to the insects. And it's my hope that we'll be able to use that information and maybe parse out amino acid profiles or understand more about the organic nitrogen in the sap so that we can get a better feel for perhaps when somebody might think that your plants look pretty good. And in your particular case, Patrick, the potato beetles come to mind in terms of a herbivore that's a leaf chewer that's actually going to be consuming leaf tissue and getting more than the psyllids will from getting the sap. You know, you have two different types of insect feeding, so there are different things to consider when you're trying to figure out how can we use this to predict who might be attacking and when. One last thing on this slide, which was interesting that Sam and I kind of discovered, is that there's some literature that shows that when iron levels increase, phosphorus and sulfur levels increase in plants, and the sap profile bears that out in this case. It's not true in every case, and this is one of those situations where some data from the field validated what's in the literature. It's a pattern that if you're creative with your eyes, you can see it in other data sets, but it was really apparent here. And that's encouraging to me that we will be able to see what's known in science in the field, in the trenches where people are depending on these plants to perform. And I'm going to add something else here as far as when I talk about. Oh, Patrick is so you can all see these comments that people are making. Is that right?
That's correct.
Yeah, potato beetles aren't an issue there. That's good. That's a blessing. But as far as the when I talk about data sets and scientific analysis and that sort of thing, everything I'm talking about from the field that I can speak to with my personal experience, none of it is experimental. It's all anecdotal, just based on the commercial grower data sets that I have. I haven't run any trials. I don't have the resources to do it. So when I make statements about what I think is happening or what I see in regard to the literature that is consistent with the SAP, I'm only saying it because it happened to be in that case. It's interesting to me, but many more samples over many seasons will tell us if there's real consistency there. And we just haven't been doing it long enough to know for sure. I don't want to mislead anybody and make anyone think that I've run trials or done anything to confirm any of the ideas I have. I have not done any hypothesis testing, but there's things that I see, and I'm going to be looking for them this coming season to determine if what I'm seeing in the STAP analysis is consistent across crops and comparable to the literature. Incidentally, as I pointed out, as the iron levels went up, so did the sugar levels, and I think that's really a result of the irons. It's an indication of the iron's importance in the enzymatic machinery that's driving the photosynthesis and the other aspects of the plant's physiology that result in making carbohydrates structural and, in this case, non-structural. I'm going to skip this slide so we can drive on. That's a squash. It has fusarium, incidentally. Now, I'll just touch on this slide briefly. This is another tomato variety called BHN 589. This is from a greenhouse that had a serious problem with the soil. We never did figure out what was going on in the soil, but the grower had a Penn State extension help him identify the problem in the plants, and it turned out to be a Pythium pathogen that was had infected the roots. And I haven't made any direct connections between what's in the sap nutrient-wise and the Pythium attack. I mean, there might be something. I haven't been able to connect the dots, but this is an example of a sap test that's completely aberrant from what we usually see in terms of the nutrient levels as they are by themselves and as they relate to other nutrients. And Sam and I are kind of optimistic that if we observe this over time, that we may get to the point where we can look at a test and say something's going on here, but it's not related to your inputs necessarily, and we need to take another look. And hopefully we can get to the point where we can do that before there are symptoms, because as you all know, when symptoms show up, there's yield loss occurring, and it's probably too late to manage it effectively and completely. I'll get as far as I can before 3 o'clock, or what time is it? Yeah, before 3 o'clock Central Time. And what you see here is a chart of SAP components that was compiled in the 80s from a couple of researchers. There are only 2 studies, but they compiled what they were finding in SAP. And I like this because if you study enough of these tests, you can take what you see in the test, even from a crop that doesn't have target values, and get an idea of what other scientists have seen with their formal studies, and you see that, yeah, those target values are pretty consistent with what researchers have found. And over on the right-hand side, I have emphasized the total low and total high values for the xylem and phloem. And what's interesting to me is that the sugars that the plant is making are not found in the xylem, and that makes sense because they're being exported. So where would they go? They'd be put into the phloem. And the phloem has a large complement of amino acids and a rather small complement of ammonium naturally. And although this says that there's no nitrate in phloem, that's not necessarily true. There's a study that was published in 2013 showing that plants can actually transfer nitrate to phloem from xylem. So we're learning a lot about the plants scientifically every day, but what was interesting is that a lot of these values are very consistent with what we see in sap tests across the board and in a number of different cropping systems. Very exciting. I don't have a lot of time left. I know we're running short, but when you're confronted with a test that doesn't have target values, that's a real challenge, especially if the grower just pulls it out of his pocket and says, what do I do? It can be a little bit intimidating. And fortunately, the handout that I prepared for you has all the notes that I had prepared for this particular section of the talk, and I don't have enough time to get into it completely. So what I'm going to do is I'll leave it because I can't cover it adequately in 5 minutes. I'm not going to try. Maybe that's a good exercise for you to study these and think about what you're seeing. I will point out, though, that, okay, I've just chopped these tests up so that they all fit on one page, but this is about a 5 or 6 week period. And if you look at the iron levels, now iron is not particularly mobile, but if you'll notice early in the season it was a pretty high level for a micronutrient like that, and it declines over the next 5 to 6 weeks. And by the end of the season, this was in broccoli, emerald crown broccoli, which doesn't have a flower, it doesn't have a fruit, obviously. So here's an example of where a crop may be using nutrients differently because it doesn't produce a fruit. So when we're studying the sap analysis, we can look for some things to be the same, like potassium and calcium. That's probably not going to vary much from what you see in a fruiting crop. But there'll be other things that are different, like especially in brassicas, sulfur and boron. They are huge consumers of those nutrients. And it's obvious because the sulfur levels they're high in this crop, whereas in another crop they might not be at this level. But you see that they're very consistent over time across the sampling period. And as Gary pointed out with the potatoes, and I never would have thought of this without his comment, the sugar levels at the end of the season again are much higher in the young sap, indicating that as the crop is maturing those florets and preparing them to bolt, it looks like there was sugar up in that young part of the plant where you'd expect it to be. But getting back to the iron, this is what got me thinking about the micronutrients. Let's see, the iron levels are going down. Why is that? Is the iron is it not available, or is it a matter of there's so much in the plant that it's not taking it up, or is it that it's being consumed and not being replaced in the sap? I don't know. That's where the in-depth literature reviews come in, and if you start doing that for every nutrient, I think the picture of what's going on in the big picture is going to emerge pretty well eventually. So what I'm going to do now is just very quickly show you a few examples of target values where several different crops are represented at the same time, if I can get there. All right, I'm going to do this the brute force way here. This is what I want. This is very interesting. Okay. Now keep in mind that the onion and squash values here were generated with the data sets that are represented here. So it's not a true validation, if you will. The tomato and pepper are, because the tomato and pepper data are from Sam's clients, as are the onion, squash, and broccoli. The difference is that the tomato and pepper standards were developed by Nova Crop Control on different samples. The onion and squash samples that are represented here are the ones that generate the target values. So some things that are going to show up that you would expect. But here are the tomato target values for calcium or potassium, and you see that these are 2 standard deviations from the mean. Most tomatoes are falling. I mean, they're actually high, but they're in that range easily. Now for peppers, there's a higher target value for potassium, which makes sense given how much they seem to consume. And Sam's clients almost invariably had plenty of potassium. That's a part of the way he helps them manage their soil. I'm pretty sure of that. Now on the onions, of course, as I said, these data sets were used to generate the target values, so they fall in that range. But you see there's not a lot of variation, especially in the young sap, in how much potassium is there. And similarly with the squash. But what's encouraging is that the squash, I mean, it actually was higher than the target value turned out to be based on Nova Crop Control's estimate. Where's that potassium going to fall for broccoli? We don't know, but we're setting trials up in Pennsylvania. I'm actually running one myself to get a feel to get these target values for broccoli in the entire range of nutrients that we test. I have several other slides on these values for different nutrients that are very interesting, but for lack of time, I just have to stop there. But I appreciate your attention and your good questions, and I think we're going to make good progress, and I'm always available if someone wants to shoot me an email or give me a call. I'm happy to help in any way I can as we learn how to use these tests. So thank you.
Thank you very much, Nick. You mentioned your the worksheet that you had, or the guide that you had created. Did you get a chance to reference that as much as you wanted to? Because we do still have a few minutes, and I think it'd be great if we could go over that while we have everyone on the webinar.
The worksheet. Which one? What do you mean?
Earlier you'd mentioned that you were going to hold off on sharing a document that you had put together.
I don't remember.
It was within the last
I don't remember.
within the last 20 minutes. Basically, okay. Let's take a 10-minute break. And Nick, I'll just follow up with you offline, and maybe we can figure out what that document was. But
Okay.
If you went to the trouble of making something, I definitely want to share that with Patrick and his group. So
yeah,
let's
I feel
take
like, okay, I'm embarrassed. I can't remember.
Okay, not a problem. Let's take a 10-minute break, and we'll reconvene let's reconvene at 15 after the hour. All right, thank you, everyone.
Do I need to transfer stewardship here, or what?
Download these items and look at them yourself and blow them up as much as you like. This is another way of looking at data. Excuse me, I haven't talked for a while. This is another way of looking at data that I like to look at, and we can determine some trends over the course of the season. And as you can see here, there was 1, 2, 3, 4 data points taken. And this is across the various varieties of potatoes, various fields of potatoes. And we sometimes will take all the data that we collect through the season on a grower, on that specific crop, and chart it against time. See what happens, to see what trends, what nutrient interactions occur over the course of the season, and as we increase one, we may decrease, how it influences the other nutrients. And this goes back to that first question that Nick was raising about sugars and what season, what time of the season things change. And these are potatoes that are in the Central Valley there in Colorado, San Luis Valley. And you've got the jellies, cucas, the Desirees, and Sangrias, and Purple Majesties. It's organic production. And as you can see here, the sugar started out way down here in the 0.2 to 0.4 range and then ranged all the way up to 2.0. And the new leaves, the dotted lines are indicators of the new leaves. The solid lines are the old leaves. So to give you a little perspective on how you read these charts. And you can draw your own charts up from any data that you collect over the course of the season on Novacrop's Benesting online website. And until you get 2 sets of data, there's no point in it, but once you get 2 sets of data, you start seeing trend, and after the third one, you can start picking things apart. And again, as far as pH staying level all the way across, relatively speaking, I suppose so. But some of the things I look at on I look for variations here and try to figure out what causes them. I don't know exactly what caused that severe drop in the pH, and I'm not a PhD, plant scientist or anything like that. But I look for outliers when I group a lot of data like this together. I just look for outliers, and this would be a question, Nick, maybe you can address for us, and what would have made that pH drop in the new growth only so drastically from before. It could have been a sampling issue, it could have been a lot of things, but the fact that it then dropped again later on a different variety here, that begs the question. All of our sampling is done prior to 9 o'clock in the morning, and that's to allow good turbidity in the leaf material before it starts dehydrating, especially in the arid to semi-arid regions of the country. So we try to take all of our samples between sunrise and 9 o'clock in the morning. EC truly does stay fairly flat. We do look at EC sometimes in regards to, we kind of use it as a gauge as to how much the ionic nutrients are being metabolized into actual plant tissues and how much is free flowing. You can run EC up with some ionic nutrients occasionally. But I'll just go through these charts and see what we can do to influence things. It's a little confusing because there's so many in one spot. I'll go back to another chart on tomatoes where I just have one variety on it, and we can look at these charts over time that way too. There's potassium and calcium all over the board seemingly, and we're having a hard time getting enough calcium. Oh, that's another thing I wanted to show you. These heavier lines, if you look at this heavy line, this solid line here, and this solid line here, those are equivalent to those two, the middle bar of the light green, dark green horizontal bar charts. That's the optimal ranges that Novacrop specifies. And one thing I've done to help me out a little bit, I made a spreadsheet, and here I highlighted the potato one. These are the optimal ranges that Novacrop has set for these particular crops down here. I picked them off of these charts. And as you can see, that's 4,000 to 7,000 on the calcium. Oops. And if you look here, it's 4,000 or potassium. I got that one off. It should be 4,000 to 7,000. That must be off by 1. So anyway, I use those for a reminder. No, that was right. It was potassium is 4,000 to 7,000, so I was accurate. I use that, and I also use that in the spreadsheet that we get back. I don't know how much you've actually seen of test data come back from NovaCrop, but you'll get those light green, dark green bar charts individualized by set, and then you'll get a spreadsheet with all the data compiled in an Excel format. So I have this one set up so that it dropped right into the bottom of the spreadsheet that you get on your email.
If I could just interject, Gary.
Yes.
Patrick,
it's hard not having any feedback from people, I'll tell you. Go ahead.
Patrick, I put samples of potato data in the Dropbox for you and your staff. So following the session today, feel free to print those off and take a look at them.
Unmuted.
Okay. So they haven't seen any sets of data yet, as far as the actual data. I should call one of those up. Let's see if I can do that without. That won't work.
Hey Gary, this is Patrick. If you would speak a little closer to your microphone.
Oh, okay. I'm not sure where the microphone is on this thing. I was trying to show you the spreadsheets as we would get them. I'd have to do that without showing you names and stuff, and I can't do that from this point. Anyway, I wanted to show you how we get data in on an email, and that would have been good for me to lay out. Maybe I can pass a screen off, pull one of those up, and show them to you later. But I tend to take the data into spreadsheets and then utilize it other ways, and you'll find ways that will work for you and your clients and make it more presentable to them, because oftentimes clients will have a tough time discerning all these charts. So rather than maybe look at this, I'll go to
Gary, can you hear me?
Yes.
Can you go back to that chart with the potassium?
Okay.
This is it. The potassium calcium ratio. Sorry. Yeah. Look at.
Yeah.
I see that the potassium is really gone down by about half or more.
That's right.
And
at the beginning of the season, we noticed he was way high on potassium relative to calcium, and we were trying to get calcium back up into his plants because he was way low. See?
Right.
And if you have potassium too early in the season, it competes with calcium uptake and thereby blocking it out.
So do you think that decrease in potassium was because you didn't add it, or because the plant was taking it out of the sap?
Repeat the question again.
Do you think that lower potassium was because you didn't add it into the system, or because the plant was withdrawing it out of the sap and using it?
Both. That was a function of both. And in order to get calcium to come back up, we have to, you have to put it in proportions. And something we've learned over the course of the season we find to be a little bit more effective is if you need both potassium and calcium, which most of the times you do, especially at tuber fill, what you can do is put potassium down one time and then calcium down another time, and let the plant take them up one at a time. Alternate shots, in either your drip or your foliars. And then the other thing you look at is your micronutrients. which with this we would scroll down to boron, your first suspect micronutrient, and I've been kind of tied up here the last couple of days, and I got kind of short notice, so I haven't gone through all of this data. But his boron was pretty decent level according to the optimal range. You can see right here between 2 and 4. So his new leaf boron was good. We were pushing boron. We were increasing boron to try to help increase potassium uptake. And you can see that the boron applications were effective. And we did bring the potassium-calcium ratio back down. Now, if you look at that other spreadsheet I had, and I kind of, some of these things, I'm not sure where they're at with NovaCrop, but if you look at this potassium-calcium ratio, you got a 4,000 minimum compared to a 1250 minimum, and a 7,000 maximum compared to a 2,000 maximum. That's a 3-to-1 ratio, basically 2.5, or yeah, 3.5 to 1. So a 1-to-1 potassium-calcium ratio isn't necessarily the case in all plants. Some plants need more potassium, just like the peppers that Nick was talking about. You'll see a lot higher potassium than a 1-to-1 ratio in potassium. In the old days, we used to think 1-to-1 potassium to calcium ratio in leaf material was a good way to go, but it's not the case in sap, because you're looking at nutrients that are flowing in the plant as opposed to what's stored in the plant. So even at that, these ratios are way high. We were clear up here to 18-to-1 on the potassium-calcium ratio, so we knew we had excessive potassium blocking out calcium, and we were afraid of the black heart coming in. Okay, so we'll just go down through. Magnesium was a little low, so we're probably low on our nitrate conversion. Sodium, not sure what the outliers on that sodium are for. He doesn't have a real high salt content in his water, but he does have some. And here you have ammonium nitrogen. And again, goes back to the magnesium is a little low, so I think we could convert nitrogen a little bit better. And here it's showing Jord has, or NovaCrop has a range for nitrate of 1,000 to 4,000. And, you know, we find keeping nitrates down is a pretty good deal. This was happening naturally. That last year was more of a sap sampling to try to determine what to do with this guy's crop the following year, and that's what you want to start out with. But what you'll find is you'll start reacting to the sap analysis over time, and one of the first things you'll start looking at is your micronutrients, because you're going to find some of the micronutrients to be deficient or excessive and have to be dealt with accordingly. But we like to maintain a fairly low ammonia and nitrate, but that's just the way we operate, and we believe that it helps with some insect resistance and getting the amino forms of nitrogen produced, keeping our total nitrogen higher. And we started coming up on some varieties and dropping down on other varieties. But I like this kind of chart to look for trends and anomalies. Sulfur, again, has to do with photosynthetic efficiency, and we've got it. We were starting to increase that, trying to keep the ammonium down. And we never did get phosphorus to come up in that particular field. He didn't do a lot of variation from what he had set done before, as far as trying to get some of this stuff to happen. He had kind of a program set. He was just kind of tracking with sap analysis to see what it would do over time. But I would say we have to look for ways to get the phosphorus in there better. Silica, we like to see higher levels of silica than even what recommended here at a top end of 35. We feel that plant health, cell wall strength is enhanced with silica, and we like to try to keep that up near the 70 range, actually. And iron is low, which is part of the problem with this phosphorus. So you can pick up on some of those nutrient interactions that Nick was talking about earlier. And Jord has a chart of elements that he shows with cations, anions, and micronutrients. And that's a great chart to look at. We have one that's been slightly revised from that even. And I think that's something I can show you here in a little bit. Manganese, a little bit low in the new leaves, but was higher in the old leaves. And we feel that you can get the manganese. We actually want to see manganese levels even higher than this. Some of what we've seen is when we get our manganese levels higher, our incidence of soil-borne pathogens is reduced. And also a lot of bacterial and fungal diseases on the leaves are reduced with an increase in manganese applications. And we have individual micronutrient components that we can market that actually the plants actually respond to it pretty well. This particular grower didn't access our organic. We have an organic form and a conventional form, but he just didn't get into that this fall. But one of the things with the SAP analysis, I think you're going to find, if you're making recommendations on products, you'll be able to check the efficacy of those products and how well they are taken up by the plants through the SAP analysis. We found some of our products, we found that we were able to get boron in quite rapidly, and actually more so than we thought we were doing. So we started backing down on some of those nutrients. So no matter what the source of the nutrient is, you can actually tell if it impacts plant uptake through SAP analysis in very short order, actually. And copper was all over the board there. Molybdenum is one of those things we find is often deficient in soils. Manganese and zinc are, well, zinc is oftentimes over-applied from previous history. Let me go back to the individual. That was some potatoes. This is tomatoes, and this is a single little greenhouse. It's greenhouse tomatoes, and this is what it looks like when you just do a single variety on those same charts. And it's a lot cleaner, and you can watch what's going on. Does anybody have any questions? I'm not going into great detail on all the analysis and things. It's more so for you to be able to know that this is a pretty nice little way of presenting data.
Gary, we don't have any questions on the side. Go ahead.
I just pulled up my sidebar because I heard somebody type, and I thought, what, somebody's sending me a question I can't see. Is this a recent question? For in-season additions of potassium or calcium, how did you decide how much? Since you don't know how far. And again, it goes back like Nick says, start out with, well, on our micronutrients, you go by label rates and see how they affect, and then kind of proportionalize on potassium and calcium. You would go by how much you have been putting down, because most of these guys are doing side dresses or fertigation. This particular individual on the potatoes had irrigation. So we would look at how much he's been putting down and realize that it's not enough, and he needs, twice as much, so we'll put half again as much on, see how much we get the meter to move, and then go from there.
And can I make a comment about that point?
Go ahead.
I have an echo, so if I sound a little strange, that's why. We had a customer doing sap testing on strawberries last year, and he was showing low calcium levels in his sap.
Hey, Nick,
He
hold on.
was using calcium nitrate.
Nick, hold on.
And one of the Keystone calcium products. It's actually an AEA product. I can't remember which one though. So what we did was I recommended he start by applying it at label rate, and we saw that the calcium didn't move very much on the next sap test. So we kind of had an idea that you can increase your rate on that. And that's what he did, and he actually just kind of creeped it up where he wanted it. And actually, with the crops that I'm used to working with in Pennsylvania, I don't have a good feel for how much a rate of any one product will move the level in a sap test. So that's something we're going to learn more about as we work with the testing and with the applications of the products. And I wish I had a better answer for you, but right now that's what we do, and we just try not to overdo it with the applications if there's a definite need. And then with the micronutrients in particular, the low rates really boost them pretty significantly. I can say that with pretty confidently. With the macronutrients, not so much.
Nick, this is Patrick. I'm pretty sure Gary can hear this too. When we switch from one speaker to the next, probably when we're done talking, if we can just mute ourselves, that way we can get rid of this echo. But Nick, I didn't hear, on our side we couldn't hear through the echo. You were saying something about a calcium product that you tried?
He was actually using two different ones. He was using it was like NutriCal, which is a calcium nitrate. It's like a zero. What is that? A 15005 or 10. I can't remember exactly, but he's using a calcium nitrate and then a different calcium product that had a different anionic component to it. And I wish I could remember, but there was some crazy situation where he had been putting on something at like a gallon to the acre, believe it or not. And I wish I could remember, but I do remember this. I said, cut your rate back on one of these products and leave the other one where it is or whatever. And we saw that it moves the potassium maybe 100 or 200, or calcium maybe 200 parts per million on the next sap test, if I'm remembering correctly. So it was trial and error in his case. But when we look at his strawberries this coming season, we'll look back on that and say, okay, here's what we did. So we'll have a better feel for what we can do this season. But in the systems where I work, there's often an emphasis on trying to get the soil biology, provide nutrients from what's in the soil. So sometimes adding something isn't always the first option. Not with all my clients, but with some. So in that case, you'd never get a feel for it. Does that make sense?
Well, on our part, the way we look at using this coming 2015 season is not just as a way of watching the plants grow, but of trying to actively manage. So we're going to need some input from those who have tried this on, like, if we need to push magnesium because we decide that's the thing to do, how much magnesium and what kind of products might we look to decide it's time to push magnesium based on the plant sap analysis. What products do we look to and how much do we use? Give us some general guidelines or tell us where to look for these general guidelines so that we can begin to flesh out a list, because it won't work for us just to use this, just to watch them without knowing we need to be able to not only see the problem but also respond to it and see if whatever response we took had effect.
Right, and that question has come up. Somebody was asking me about that last year in regard to tissue testing, and I told him at the time that I wasn't aware of a matrix or anything where you could go there and look at a particular product formulation and get a rate and then have a prediction of how that would change what was in the tissue analysis. So I understand your point. That's not exactly what you're asking me, what I just said, but I hear you. I know there's a need for that. And as we proceed this coming season, I'm keeping this stuff in mind because there's things we need to do with our people, not just with Michelle's other clients. So I'll be making notes and trying to find out what I can and learning as I go. Like I said, there's so many questions that you ask yourself when you think you found a single answer that there are rabbit trails everywhere. That's not an excuse, though. So, yeah, it's one of my goals is to help answer questions like that.
I agree with Nick on that. There's no set prescription on so many pounds of actual potassium applied is going to respond in a sap by this many parts per million. There's no correlation there yet. And the thing is that we find that something like potassium is pretty quick to respond, but calcium's a good bit slower. We found some issues with trying to get phosphorus in the plants. And we tried all different forms of phosphorus, couldn't get it to go in the plant, and the plants were still suffering from it. And actually found out that there was a bit of a chloride conflict. And once we, well, that was towards the end of the season, and reviewing, looking at these charts, we found that we had what we thought was excessive chloride. Let's go down here. I think it was on this particular farm here. And maybe Nick, you can jump in here. But you said chloride's necessary in the plant, and we understand that. And the question is by how much?
Yeah.
And,
yeah.
And well, come on here. Here it is. And we're right in the middle of the range, but we felt that 1400 on the new leaves as it was going up, these plants really started to get sick back in this period of time. And we were having a hard time getting phosphorus up in these plants. See, we never did get the phosphorus up. And they're one of the anions that compete with, chlorine's one of the anions that compete with phosphorus. And maybe now would be a good time to show that chart that Jord talked about. And on this chart, you'll see it's divided into 4 quadrants. This is a little slow response. And you got your cations. And anytime in the cation quadrant, if you've got too much of one of these nutrients, you're going to have less of the others. And with chloride and phosphorus, we had a higher chloride. The phosphorus was low, and I believe our sulfur was low too. So we wanted to, this year, we did some situations with the soil in that greenhouse because these were planted in the soil. And we checked his water, and he was getting chlorides from his water, plus he had some residual soil buildup of chlorides. So we tried to mitigate that this winter time, and now he's replanted into that greenhouse, and things are looking a lot better on his regrowth of this new crop season. So that's one of the things that even though we were in the optimal range, the chloride was the highest thing in the anions that was working against us. And right or wrong, we grew some cover crops in there over the course of the winter, and the cover crops look a lot better now because of the fact that we got some of the chlorides mitigated, and we're treating as water to keep the chlorides out. So we go into more than just nutrient applications. We're going into water treatment as well, which is common in consulting business. Is there some questions? Doesn't look like it. This chart here, I can put that in the Dropbox too. But this is a pretty handy little chart to keep you appraised of what's going on. And also diagonally across this chart, you'll see some interactions like iron and phosphorus, boron and silica, and things like that affecting calcium. And this is a good chart, kind of like that plant chart that Nick showed early on in the session. I like to keep this one around for showing the customers. It's more of a visual for the customers to help them keep track of all these nutrients. Manganese we found on the low end, and we found a few things, and you'll find your own little things that happen in your soil types and your management practices, I think, over time. And as we bring up manganese, I think it helps with potassium modulation or regulation, and there's a good bit of information about viral and bacterial diseases, fungal diseases on leaves. And this is one of the things, I walked into the potato fields up in Minnesota, and they also have potatoes in Arizona. And before I ever took the first sap analysis on that particular farm, I looked at the plants and kind of felt like there might be a manganese issue with some of the viral problems they were having, and actually seeing some manganese deficiency in the leaf symptoms. And sure enough, when we got our sap analysis back, we did have that. So that's another good way of having the sap analysis, good for verifying some of your suspicions, because oftentimes there's more than one nutrient that's impacting the health of the plant, and it looks like one, but it could be a combination of two or three. So I like this set of charts just for kind of the long range, end of season, well, throughout the season. When you see nutrient lines cross, this is in copper, it wouldn't be so applicable in copper, but if you're doing it in potassium, and you got those lines across, here we got the calcium. That's an immobile nutrient, so it's good if the you have to supply calcium throughout the season. You want to keep your new growth calcium up. It'll tend to generally increase over time in the old leaves, but it's maintaining the calcium in the higher level in the new growth, especially in a continuously fruiting product like tomatoes and or potatoes, being that they put on 2 and 3 sets of buds. Let me see. Here's some of the potatoes I was looking at. We just got started with this farm, and started with sap analysis in August of last year, and wanted to see where they were at and see what needs to be approached for this year's production. We consider their potassium to be considerably lower than we'd like. Seldom do you see a crop. This is the only crop that I've seen actually last year where we had more calcium than we had potassium. So they were running out of potassium when I showed up on the farm in August. And so we're going to have to do something about keeping their potassium levels higher, because I don't know that I've ever seen calcium higher than potassium like in this field. Magnesium was considerably high. Sodium's okay. They had almost no inorganic nitrogen, and their total nitrogen was down, so they weren't getting full photosynthesis. Now, this is a seed potato farm, so they're not trying to push for size. They just want a large volume of buds, number of buds, and of tubers. So they're not actually trying to grow in size. So I think they're trying to hold back their potassium, but this is telling them they're holding it back too far. So we're going to try to manage that for them closer. Chlorides seem to be extremely high for my situation. I don't like to see in the 4,000s on chlorides. And as a result, you'll see that it competed for phosphorus and reduced their phosphorus uptake. Sulfur is high for some reason. I don't know what that is, but I wouldn't want to see it that high. Again, iron is low, helping reduce the phosphorus. At the same time, they're being outcompeted for phosphorus by chloride. So they've got two things working against their phosphorus uptake. Manganese, those levels are okay, but I kind of like to see a little higher even than out here. We'd like to see probably up in the 20 to 25 right now is where we're targeting to try to alleviate some of the potato virus, curly top, and fusarium issues.
Hey, Gary?
Yes.
On these interactions where you've got, Michelle, can you mute Gary's just for a moment?
Oh, I'm sorry.
Yeah, we're getting an echo, so on these interactions where you've got phosphorus and chloride and these other anionic nutrients like sulfur, and I'm guessing that's sulfur in the sulfate form, or is it sulfur in the elemental form?
I think it's probably, I'm not as certain, but I know that's represented by sulfate ions, and maybe the sulfur in sulfur-containing amino acids or other sulfur-containing compounds, but I don't think it represents elemental sulfur.
Okay. So on these anion antagonists for phosphorus uptake, if phosphorus uptake was somehow limited by chloride, is it possible that other anions, any other anion, could be competing with phosphorus, or is it specific? Like if there was an excess of nitrates, for instance, could you affect phosphorus uptake, or some excess of sulfur in the sulfate form? Could you affect phosphorus uptake, from Gary, from your experience?
Gary, you'll have to unmute to talk again.
Right. There you go. Now, I tried it once, but you must have hit me one more time the same time I did last time. According to what Jord would indicate, and in our experience as well, anytime one of these elements is high, it'll make it more difficult for the other three to go up. If two of them are high, the other two will be low. A high chloride means generally that the other three will be lower. Now you asked about the sulfur, so let's go back and look at the sulfur. It's high and the chloride's high, so that's two of them high. So phosphorus is low. It's a balance. Jord puts them on like a teeter-totter, and he puts all those 4 anions on the teeter-totter, and if one goes up, the other 3 goes down. In this case, you've got 2 of them that are high. Nick, you might want to address, in my opinion, 630, and if you look at this optimal range here on potatoes, sulfur should be 175 to 300, and you're clear up into 638s.
Yeah, if that's what they recommend, then it's high. for sure. But my question is why? Where's the excessive sulfur coming from?
It could be coming from sulfur applications to fight bacterial diseases or whatever they might be using it for. I don't know. I'd have to look.
Gary, you're muted. If you could unmute yourself if you're talking.
There you go. Sorry about that. This is the potato program that they were using. It says 2013, but they had used the same program in 2014. So then you start looking at what they were putting on and what we're going to do differently next year. So here they got 250 pounds of SOP, and they also got a AMS in there, so they've been putting some on. And the question is if they've been putting too much on. Whoop, that's not what I wanted. This is probably driving you nuts, I know. That's one of the things as a consultant, when you see something that's an anomaly like that, you have to ask, okay, what did you do to provide, what's the source of your chlorides? In the tomatoes, we couldn't figure out, because he hadn't applied any chlorides at all. So finally we tested his water. The chlorides were coming from the water. If they use much potassium chloride in their history, you can have a buildup of the chloride in your soils. And he had some chlorides in his soils in the tomato fields. And these guys here, we just said no more potassium chloride, period. And they've been using some in the past. Sulfur, we'll just have to not put a lot of sulfur on this coming spring and watch our first test when they come out. We didn't do anything with nutrition last year on this crop. We just got called in at the very end of the season, and they only had time to take 2 different sets of data before they actually harvested their potatoes, because they didn't find out about us until August. Any more questions on that? Did I answer your questions as best I could, I guess? Okay.
You can go on.
Say again?
Gary, go ahead.
Again? Gary, go ahead. Okay. So we get into the micronutrients again. I'd like to see the manganese even higher than what it is, and molybdenum is almost non-existent. We find that molybdenum is usually almost non-existent in almost all fields across the nation as far as all the crops that I've dealt with. And manganese will be second in line for that normally. And we find if we can start getting that up in the manganese, our crop health starts improving quite rapidly. And other things start to come into line, and I can't explain the interactions between that. But when we start addressing these micronutrient deficiencies or excesses, we can start turning crops around, and the macronutrients actually start to balance out a little better. And here you got a little higher ammonium. It doesn't even show up on George's chart as far as high, medium, or low. Ammonium, the way I understand it, can be converted fairly quickly in a plant if it has a good, healthy photosynthetic process going on. And molybdenum is one part of that. So we like to keep the molybdenum up to keep the photosynthetic efficiency up. You got plenty of magnesium. Yes?
On molybdenum, how do you go about fertilizing or how have you approached it in other crops when you saw it was deficient or you couldn't find it in a test? What products do you look to?
We have our own products for that. We have an organic line as well as a conventional line, and that's one thing we have found that they are very efficient at getting into the plant. We usually do it through a foliar. It can also go into drip line and works very efficiently. And a pint to the quart to the acre, I think, or that one I think is 4 ounces actually. It's it's one of our own products though.
Okay, thanks.
Generally speaking, generally speaking, the micronutrients we feel need to go in as a reduced state as opposed to an oxidized state nutrient. So we try to use micronutrients that are supplied in a reduced form. Okay. Well, that's what I got there. I think that's pretty much what I've got for right now. This is most. I just tried to give you what I could on potatoes. Here's another potato field, and here the potassium's deficient actually because it's running out again, and calcium's not. It was high very early on in the season, and then it's dropped off later in the season. And now you see instead of being a 0.7 to 1 potassium-calcium ratio, which is the first time I ever saw that was in the other set I showed you, this one's a 3 to 1. That's more what we would expect to see for potatoes. And in this particular variety, he's still high in magnesium, way high in magnesium. Sodium's okay. He's high in nitrate, what I would call higher in ammonium than we'd like to see. And total nitrogen is still low, so he's not getting good protein synthesis, amino acid synthesis. And so now here his chlorines or chlorines are okay. Sulfur's better. Everything's a lot more balanced in this particular sample. And you see his iron's up some, his phosphorus is up some, manganese, and still yet his molybdenum's still way short. And that's what you'll see most often. Any questions on that? Nick, do you have anything to add?
I'm sorry, Gary. My attention was distracted. I'm sorry. We were talking about these potatoes. This is a potato chart here. Can you all hear me?
Yes, it is, Nick.
Okay. There's a lot of. What's the magnesium source here? There's a lot of magnesium.
Well, like I said, this is last summer, and we didn't do anything with this. I had showed what they had used for their nutrients. And basically, they're just in high magnesium soil. That's another thing where they're at in Wisconsin or Minnesota. it's high magnesium soil, and that's one of the things we find out there in the West is quite a bit. Is a lot of high magnesium. So we got to work against that all the time.
Well, what's interesting to me is that the ammonium levels are lower in this chart than the other one.
Slightly lower, yes. And we really try to manage. I got to make sure I get back to the right one. We try to manage with minimal amount of ammonium and keep that fairly low by a good photosynthetic system operating. We find in the better, healthier growing plants, oftentimes, not always, often there's nothing always, I don't think, but oftentimes the ammonium levels are low, especially in the morning. We sample everything between sunrise and 9 o'clock.
Yeah, there's definitely a photoperiod effect on the nitrogen levels, but I've drawn a blank on exactly how it's represented on in terms of the levels of the different forms of nitrogen. But the light-dark cycle has a big part in what is present early in the morning with both carbon and nitrogen. And those are the sort of details I read it and read it and read it. I can't remember.
Nick, I think it's the nitrate accumulates during the dark, and then it gets cleared out in the ammonium or in the protein when photosynthesis kicks back up. So that you're not picking up high nitrates in this early morning sample suggests that there shouldn't be a big pool of nitrates waiting for you, even during the daylight hours and later on in the day.
Right, because does the plant process them at night with this, by using the starch that it's stored up? Is that how that night cycle works in the dark?
Well, the plant continues to respire and transpire, so the going from nitrate to ammonia and then ammonium then to an amino acid is a reaction that requires light energy.
Right, yeah, of course.
The nitrate carried in the transpiration stream, so that little bit of transpiration that's happening through the night is what's charging the vascular tissue with more nitrate.
So it can actually reduce the nitrate at night.
Yeah, so that's why on petiole sampling there's often a caution given that don't pull too early in the morning. But if it's not becoming structural and it's not getting into the leaf tissue itself, it's not being shunted into the leaf, then maybe the plant sap analysis isn't even looking at it anyway.
Right. Now, Gary, can you go back to that other one, the prior one, so I can see the zinc? It's still good. It's just a little lower. I know I was reading somewhere recently that zinc is, I mean, there are a lot of things important for photosynthesis, but zinc in particular is implicated in optimizing it.
Yes, zinc will enhance the photosynthetic process as well. I can't remember, maybe we were on the same call or something when we got that information. One of the things I find interesting here is that the phosphorus is still low even though the zinc is high. Generally, zinc helps with the uptake of phosphorus, but that is blocked out by the chloride and the high sulfur, so that makes it even more difficult to get the phosphorus up. Come over here, zinc is a good bit higher yet even, and there your phosphorus is up.
I have another note here. Notes that when zinc is deficient, protein synthesis is impaired. And that's mainly because the proteins that need zinc to carry on photosynthesis start to disintegrate. So that's the sort of the clues I look for in my research, that when I do my literature reviews, and try to match them up with what I'm seeing in these tests. And then the next question, as I was saying in my presentation, was what are the implications of the SAP movement for that? So if we can answer questions like that, then maybe we could eventually get to the point where we have some predictive capability. But you don't know everything that's going on outside the SAP test results, so it's a challenge for sure.
That it is. And if you guys, I don't know if you've had the opportunity to view the SAP seminar recordings that we took in Santa Clara or in over in Ohio. Last year we had one in March and one in November, but if you listen to that 10 hours of video there, you'll see you're going to some of these nutrient interaction discussions, and some of them are a cascade of 4 nutrients before you get down to the causal one. So those are quite intriguing, and they're developing and discovering new interactions with the more data that they get coming across from different crops. And the ones that he talks about, like what we're talking about here with the cation antagonist and the anion antagonist, and then the micronutrient interactions with those major cations, all of those he develops over time across different crops, and those interactions are congruent throughout all the crops, regardless of which species we're talking about. So that's one of the things that I like about the SAP analysis, is it doesn't matter what crop it is, the interactions are very, very similar in most crops. The levels may be different, but the actual relative interactions are similar. Michelle, was there anything else you wanted me to add to the discussion here at this point?
No, Gary, I don't have anything additional. We do have some more content that Nick has generated, and time allowing, we'll turn the stage back over to him. Some worksheets that he's prepared. But I want to give Patrick the opportunity. Patrick and your team, do you have any specific questions that you haven't had the chance to have answered?
I'm going to open it up, and anybody in the room here with us can just ask their question directly to you. So,
hey, Gary, you said something about that they were managing a seed farm to have lower potassium in an attempt to get bigger sets.
I'm not saying that they were probably not as concerned about potassium for tuber fill as a fresh potato grower would be, because they didn't want size. They wanted to maintain smaller size. They just wanted more buds and more tubers, and that's one of the assignments they gave us, is to stimulate more tuber formation and maintain those early set tubers. Nick alluded to earlier that the early sets are critical to maintaining potato yield, because the potatoes will set the first set and then they'll go back into vegetative state and then reset another set later, and oftentimes the initial set sometimes may atrophy. So what we want to do is stimulate more tuber set and maintenance, and they're still going to require potassium regardless. But that was a thought that I had, was that maybe they weren't trying to feed potassium as hard because they knew they didn't want too big of potatoes.
I think Yes is wondering.
Hey Gary, on following up with that question that it was just asked of you, what are, from your opinion, what are the critical nutrient interactions early season that could influence that initial set formation?
Okay. This gets into some of our own philosophical stuff, I guess, and maybe not as much scientific research, but more anecdotal, I guess, through experience. And we're finding there's some more than just nutrients, then there's also hormonal shifts that occur at certain phases of the plant's life cycle. And there's certain elements that we can use, and we're looking at using some cytokinin stimulation to stimulate more aggressive flowering, tuber set, and then maintaining calcium intake early in the plant life to help with cell division stage of plant life. And of course, we'll need to keep our phosphorus up. The phosphorus being low here, I think, is one of our key things. And cytokinin.
So for this potato, how early will you be able to take a sample to know if you're starting off well or if you're starting off way far from where you want to be?
Well, interesting you should ask that, because one of the key elements we want to get into is get that lab over here in the United States so we can test the tuber before we stick it in the ground. And then after that, you can, a full set of sample is young leaves versus old leaves. But we can also do a half set and do young leaves only, and that would get you a beginning start. But generally speaking, on potatoes, most of the nutrients in that new growth would be coming from the tuber itself, from the seed piece itself. So we like to see as early as possible. If you're really jumping to the gun, you can do it as soon as you got emergence. George was asked that question back in March of last year, and somebody asked him if he could test buds on a tree fruit. Just the buds just coming out in the spring, and he said if you want to collect 75 grams of buds, you're welcome to it. And they could actually test those buds and see what those buds are coming out with at that point. Now, how much value that had, I don't know. But you can do a young leaf as soon as you got a leaf that's fully up and photosynthesizing is the best way to go. I have some sets of samples that came in that have really messy numbers on them, and I think it was because the new leaves were just picked too new. They weren't fully developed and photosynthesizing yet. And you'll see some instructions on the sampling guides that will help you pick the right leaves to take on your sampling. And that's very critical that you're very consistent with that from time to time.
In the Netherlands, have they gone to this tuber testing method for pre-plant testing of the seed?
I can't answer that, to be honest with you. George, I know, does test some fruits and vegetables, and what they do with it, I'm not too sure. We have a concerted effort or interest in doing so because our whole company vision and mission is to grow better food, basically, and we're looking at this as an opportunity to possibly quantify quality of fruit, grains, vegetables, whatever. So that's going to be something we're going to explore more when we get it over here, because we can't ship potato parts overseas over the customs borders and get the results back.
Michelle, can you comment on how far out the lab construction is and when the U.S. lab will be able to receive its first samples?
Yes, I can. The lab project currently is.
Gary, perhaps you know.
Oh, can you hear me now?
Yes.
Okay, perfect. So the lab project is still underway, and what we are doing right now that I can share is evaluating the options for various investors who we have available to us, just looking for the correct partner, if you will, or um, the partner that we see the most potential with and that we are most philosophically aligned with. Our initial goal was to just bring one lab to the States, and that has expanded in recent dialogue, and especially with some markets that are available directly in and Colorado and California. We're exploring all options there also, and I can speak to you more on that, Patrick, offline. But in terms of time frame, I don't want to overpromise and not be able to deliver because there are a lot of parties and a lot of moving pieces. But our goal is to have the lab operational by the end of 2015 and be using it here in the States in 2015. And until then, we're still going to facilitate the shipments and the dialogue to the NovaCrop lab.
Okay, thanks.
Sure.
I want to thank you all for your attention and questions as well. And if I can be of any assistance, I am planning on coming out to Colorado the third week of March, I believe. And either prior to or after that, I'll be out there for several days. But at any rate, I do travel through Colorado on a pretty regular basis. Have quite a few customers out there and getting more as we speak. So I'd be glad to meet or visit with you. If you would like to call, that'd be fine as well.
We appreciate that, Gary. And yes, we do want to make that opportunity available to you, Patrick, and your team there. That we had initially intended to have Bill be able to sit in on this presentation today, and schedules did not align for that. So we still extend that offer to you, and Gary has indicated he's also available for some on-site training for your staff.
Great.
I actually have appointments all day.
Nick, what I'd like to do, Nick, is allow you the opportunity to go ahead and share what you had mentioned earlier, and we were a little stretched or a little pressed for time. But if you could go ahead and prepare that content, and we'll turn the screen over to you.
Okay. I'll get that up.
Is there something I have to do here, Michelle, to transfer it over?
No, Gary, as soon as Nick's indicating he's ready, then I'll switch the screen over to him.
I'm ready whenever you can go, Michelle.
Great. Go ahead, Gary. I'm sorry. Go ahead, Nick. And then the rest of us will just mute out.
Okay, so excuse me. This is at this point it's where it gets a little more academic because what I'm going to talk about some things that I've found that may or may not be something that you can use to make a recommendation, but they are important aspects of nutrient interactions that I think we'll be able to discern from the SAP analysis. And over time, as we see that these patterns occur consistently, you can be confident that you might be able to get 2 or 3 weeks out from a phenological stage or from an important event in the crop cycle and say, okay, this is what we can expect with a reasonable amount of certainty, even though it's not going to be confined within confidence intervals or any sort of statistical certainty. And I'll just tell you some things that I've found out as we go and outline some nutrients using an example from a broccoli patch where we don't have target values because we don't have target values for cole crops yet, and by the end of this season we will. This is a grower that is organic, doesn't use any pesticides, and is pretty intensive with his nutritional management. He uses a lot of the products that Gary markets, and he doesn't really spare any expense to do that. So if whatever his consultant is Sam Zook, who runs Keystone Bioag, and if Sam tells this fellow to do it, he'll do it, but he also does a lot on his own because he's pretty sharp, and he's learned a lot himself from doing these tests. So one of the things that is known about manganese, since that's a pretty important micronutrient from a disease resistance perspective, it's well known that manganese can have a really positive effect on the plant's natural defenses against some pathogens. But there's some other things about manganese that are rather interesting. And one of them is that manganese deficiency can have a detrimental effect on non-structural carbohydrates, which basically in our situation would be sucrose. So in other words, if manganese levels are low relative to that crop, you may see low sugar levels too. There may be low sugars because of something else, but the plant physiologists have determined that low manganese can contribute to low sugar. So if we look at this test in front of us right here on May 19th, if you look at the manganese, it's about 2.5 parts per million in the young sap, 2 parts per million in the old sap, so not too much different, which is what you'd expect from a nutrient that's not particularly mobile. And if you look at the data from the 29th of June, you see that the manganese has increased by more than almost doubled in both of them, and the sugar levels have also increased at that stage of the game. So the plant is really producing sugar and exporting it, and the manganese levels are really good in both young and old sap. So if there is any mobility to it, it's really being transported where it can be used. Now, other indicators of good photosynthesis would be all the micronutrients, and I see on this test that, well, I don't see a huge difference from that first test to the last one in terms of increases, except in manganese, because the iron levels are lower, the zinc levels are lower at that stage than they are at any other point in this testing cycle, and the copper is also hasn't really declined steadily but is lower. and the molybdenum is higher. And Gary did mention that molybdenum is implicated in photosynthesis, so that may be having a role too. But my point is that in this particular case, it looks like the observation about manganese and sugar is confirmed here, and at least in this sample. Will that be the case in the next one? I don't know. But as I keep saying, finding these patterns consistently can build your confidence that you might see that moving forward on other tests. And I personally am encouraged by that, because these tests are bewildering if you don't have a starting point at all. Does that make sense?
The last comment that you made certainly made sense. If we don't have a starting point, they are bewildering, yes. On the manganese deficiency, or something less than a desired level, having effect on non-structural carbohydrate development, has that been applied to forage production? That's actually a component that's sometimes tested for within forages, is non-structural carbohydrates. Do you know if people are actually actively managing pastures or forages with manganese inputs, manganese foliars, to influence that?
I personally don't know, because I don't do a lot of work in forage systems, but Gary probably will know better. I think Gary sees a bigger variety of different things than I do. Is that, Gary? Do you know anything about that?
We are generally increasing manganese levels over and above what even Novacrop's optimals are. Now that's just by our choice and by our observations, and we see it in particular helping against disease pressure. As far as how that impacts the non-structural carbohydrates, I can't address that particularly, but that's something I'm going to start looking for. I made a note of what you were talking about there. That was the first time I've heard the manganese being associated with sugar production. And I just looked at some of my other charts just to see if it varied. And I didn't see where we had a deficiency that sugars got lower, but I didn't have any, severe deficiencies. But I was looking for one that we got the manganese higher on to see if it increased sugars at the same time. So I'm going to continue to look for that. But we'd like to see manganese levels. We'd like to see them as high as 20 to 25, actually. You can get a toxicity.
Yeah, and that's
a pretty high level toxicity.
Patrick, back to your original question about alfalfa. You said alfalfa, right? The information I just shared with you about the manganese, I'm pretty sure it was determined on some kind of a C3 plant or dicot, so it was not done in corn. It was done in something that has the same vascular system and general physiological underpinnings as an alfalfa plant. But no, maybe if a legume might be different, in that respect. I don't know that, than something else that's not a legume. But speaking of manganese in terms of its toxicity, Gary brought up a good point, and I have another note here that says manganese toxicity can occur with induced deficiencies of copper, magnesium, iron, and zinc. But if you have adequate magnesium, you can increase your manganese input significantly. And generally, the SAP analyses are showing us that our magnesium levels are consistently good, if you will, that the sap has an adequate complement of magnesium for the plant's needs. because we don't see them change very much, which is indicating to me that it's not being moved someplace because it's being consumed or the plant's losing it somehow. And here's an interesting connection between manganese and indole acetic acid, which is a growth hormone. If manganese levels are high, in general in a plant, not just speaking about sap, if they're high or excessive, indole acetic acid will be degraded. I don't know the process that causes that, but it's related to the enzymes that break down indole acetic acid, obviously. But what happens is indole acetic acid, it's an auxin. So it's being transported from the meristem and the young growth down through the phloem. At the same time, calcium is being transported through the xylem towards the top of the plant. And there's some sort of interaction. Maybe there's a communication between the xylem and phloem. I don't know what it is exactly. But when the indole acetic acid concentrations are lowered, calcium transport is impaired. So the relationship there is a high amount of manganese can drive down calcium transport. Do you follow?
Yeah, there's a few of us writing these things down because they're new. Haven't heard them before, so give us just a moment. Hey Nick, can you review one more time this manganese toxicity interaction with the 4 nutrients you listed off? Can you go through that one more time, please?
Yep, roger that. This is one of those interactions that I just talked about in passing the first time I was speaking, but manganese toxicity can occur when there are induced deficiencies of calcium, magnesium, iron, and zinc. So there are interactions between 2 cations and 2 micronutrients with the manganese in that case.
So it's calcium, magnesium, iron, and zinc. Is that correct?
Zinc, that's right. If those are low,
copper's not involved in that?
not according to the source I had.
Okay.
So the take-home message there, and it was a point I made with the magnesium, is that if you're operating in a system like Gary and I often do, evidently, where magnesium levels are adequate and the plant doesn't have a requirement for that based on what it's moving through its sap, you can increase manganese levels considerably. And that's probably part of the reason that the inputs that Gary's crew is making are having a good impact without being overly toxic.
Okay, thanks for clarifying that.
Now here's another one, Patrick. You'll like this. There's an enzyme called cytochrome C oxidase. And if I remember correctly, it's actually like one of those enzymes that scavenges reactive oxygen species which are damaging to cells. I think that's what it is. It's a cytochrome. But one of those enzymes requires 3 copper ions, 2 iron ions, a zinc ion, a magnesium ion, and a sodium ion. So that's actually 6 separate ions and 3 copper of those 6. So half of it's copper. Now that gives you some idea of the kind of micronutrient load that a plant needs for its important activities. And when you look at tissue samples that have been collected over the years in different crops, and they've kind of outlined what's considered adequate and toxic from a percentage dry matter standpoint, it's always like in the thousands of parts per million when you convert them. And you can convert percentage to parts per million by multiplying by 10,000. So the levels we're looking at in the sap are many, many times lower than that in parts per million if you compare them unit to unit. So there's another clue that those two bits of information together should indicate that micronutrient loads in sap can be pretty substantial before you reach a level that's considered toxic, although that doesn't give you free rein to go dump it on.
Okay.
I'll put myself on mute if somebody wants to make a comment.
I think we're okay on our side.
Okay, so what I'm going to do now is just change gears a bit and go through some of the notes I made on these Emerald Crown slide test results. They're not necessarily related to what's in the test result, but it's information that I felt I would use if I didn't have target values, and it's based on what is known about plant physiology. So some of this is going to be almost review at this point, like this stuff. So we're not gonna harp on that too much. But one thing that's in my mind is important regarding the relationship between nitrate and ammonium, and Patrick actually alluded to it when he was clarifying something for me, and that's that I think that I'm going to see this, and I just started looking for it this winter, that as nitrogen levels in the sap decline, that is, as the plant's consuming the nitrogen, you will see increases in ammonium nitrogen, because ammonium is ultimately the reduced form of nitrate. In a sense, it's a reduced nitrogenous substance that comes from nitrate. And in this particular data set we're looking at right here, you can see that that's what happened. And when a plant is operating at its optimum, I think you'll see that across the board. And these Emerald Crown broccoli were a beautiful crop from what I understand. So that's an indication that you can expect that sort of academic stuff to be borne out on these practical field-based tests. And I might add that, excuse me, at the beginning of the season here, the nitrate levels were rather high in the sap, and this goes back to something Gary mentioned about young plants that aren't equipped with the machinery to process nutrients as efficiently and as quickly as they are when they're in, say, their full vegetative form. And that's, I won't say exactly, but I'm pretty sure that's what was going on here, because the nitrate levels in the sap, obviously it's still consuming nitrogen from the soil, but it's operating at efficiency, so it doesn't show up in the sap.
Hey Nick, I got a question for you.
All right, hold on, I gotta mute myself.
On the part of tomatoes, how tall is a tomato plant before you would pull your first sample looking at top of plant compared to bottom of plant? How tall might that plant stand?
The growers that I work with will start sampling their tomato plants about 4 weeks after they transplant them, because in Pennsylvania, invariably, I mean, basically 100% of the tomato growers will start them in greenhouses and transplant them. But some of the more advanced growers that are using sap analysis will start sampling before there's a young and an old leaf, I don't think that's the most useful thing to do, but it doesn't hurt. I would rather wait till they have an old and a young leaf. So 4 weeks after transplant, if they're big enough.
One thing, if I might add to that, one thing you want to consider is where's the source of nutrients coming from that are going to give you the results you're looking at. So if you're bringing in a transplant, he's going to be getting nutrients from that plug. He's going to be getting nutrients from wherever he was fed beforehand. So you got to take that into consideration when doing so. And then if you're going to do an application after that, I would probably start with a foliar first and see how then that responds at the next set of samples. But just take into consideration whether, on corn plants, for example, they want to take a sample as early as they can. So they go out there and we don't like to see the cotyledon because that's all coming from the seed. But if you take the first true leaf, you're getting farther away from that cotyledon stage. But technically, you could take it from the cotyledon, but that would tell you what was in the seed more so in the soil.
And I might add to that comment about the cotyledon that cotyledons tend to be deficient in ammonium, if I'm not mistaken. So they may not show ammonium at all, which could give you a reading that would seem strange later. There's one of the inorganic nitrogen forms that cotyledons don't have in them, but I think it's ammonium. If I'm wrong, I'm sorry. I don't mean to give you bad information. So, well, actually, does anybody else have a comment before I go on? Okay. So, yeah, we don't really need. We won't comment too much on that, other than to say that, as we saw before, much of the total N in a plant that's actively photosynthesizing and actively processing nitrogen, most of that nitrogen will be in the form of amino acids, polypeptides, and proteins, and this data set is bearing that out. And what's kind of neat is that early on in May, you see that you see the transition happening over time. So, getting back to one of Patrick's comments about, well, we don't just want to watch what's going on, we want to have something that's actionable. The more of these tests that you run, you will start to get expectations of what to see, and if you don't see that happening, say in the first 2 or 3 weeks of sampling, or 4 weeks, then you can take action to correct it for the next test. But that only comes with time and the experience of running the tests. Okay, there's some stuff about sulfur. In Pennsylvania, our soils tend to be deficient in phosphorus, and since it doesn't move very well, and the state of the plant's root system is critical for phosphorus acquisition. But one thing that Keystone Bioag is going to try this coming season is emphasizing the more use of mycorrhizae in the root zone to try to improve the phosphorus availability. And I think that's a good idea for a number of reasons. Now here's something interesting about iron that I've been wanting to talk about, but didn't really have a good platform, but we do here in a sense. There's an enzyme in a plant system called glutamate synthase, and it requires what's called a ferrodoxin. The ferrodoxin is a conglomerate of iron and sulfur. And when glutamate synthase has its iron and sulfur complement that it needs, it will make glutamine. And when glutamine synthetase, which is another enzyme that converts glutamine, has adequate manganese and magnesium, and ammonium, it will make amino acids. And that's standard across plants. What's my point? If ammonium levels are persistently high across, say, in a long crop like tomatoes, where you could sample easily for 12 or 14 weeks. If you're seeing ammonium levels that are persistently high, if sulfur and iron are amended, if they appear to be low according to the test in the sap, there's an indication that there's not enough of those two elements in the sap to be going to the cells where they can form the ferredoxins to help convert that ammonium. Now, I'm not saying that's exactly what happens, but a lot of what we do is guesswork in agriculture, let's face it, and that to me seems like a good guess, something to try in a situation like that. And what's interesting about this Emerald Crown data set is that the sulfur levels are pretty consistent across each week, which you would expect in a coal crop. And as I told you before, the iron levels were slowly creeping downward. So you would think that iron was getting deficient. But that might not necessarily be the case, because the ammonium was being converted because it was staying low. And the total nitrogen was staying at a level that was sufficient for this plant's needs. And nitrate was being converted and turned over. I would conjecture that in this case, without knowing the target values, that the iron levels were actually sufficient in these plants at those levels. Now, I mean, you see a spike in ammonium on the 17th of June, but that was a sample that got held up in customs. If that had not happened, I would have expected it to be lower. So I'll put myself on mute.
That's fine.
Molybdenum would have an impact on that as well. And you look at your molybdenum levels, you're from 0.43 to 0.176, 1.76, and your ammonium continue to go up. That isn't right then. Magnesium, sulfur, and molybdenum are three of the major ones involved in that, as well as the iron.
Well, Gary, there's something else to remember about ammonium. Ammonium can be created from several different processes in the plant, so we don't know what's considered high in coal crops, right? So it's possible that that plant was metabolizing things that were, maybe it was breaking down proteins for some reason, which would have contributed to the total nitrogen pool, but also to the ammonium pool. And who's to say that molybdenum wasn't effective with those other nutrients and driving that in a direction that the plant wanted it to go anyway? Is that right? And I'll also mention that these plants did not show any symptoms of ammonium toxicity, so that's another indication that these levels are in the sap aren't indicative of a problem at this stage, at late in the season. Now, early in the season, like we said before, the plant's not as active, so that might explain why they were high there. Now, in terms of, let me see where this is going next. What's the next nutrient here? Okay, we talked about this. All right, I'll put myself on mute. Hold on.
Okay, on the ammonium toxicity, what would that look like on a plant?
Good question. Now, I don't know what it looks like on coal crops, but if you take, I've seen pictures of it. Let's say a tomato plant. The plants will they'll start to get stunted, and they'll turn yellow because the ammonium is. I'm drawing a blank on how exactly the toxicity manifests itself in the plant, but the symptom will be a stunted plant. Obviously, if it's not processing ammonium and making amino acids, it can't grow, and it can't photosynthesize properly. So that's part of the yellowing. Now, one interesting problem I saw in a greenhouse just this winter with ammonium was a calcium deficiency that was showing up as necrotic black spots on the edges of the leaves and puckered leaves on the upper parts of the plant. And I saw that, and the guy who's growing the plants told us that his soilless media, it's actually like a compost that he makes. It's high in ammonium, and I saw the tests for it, so I could attest to that, and low in calcium. So that was so severe in these plants, which were like 6 weeks old, that the top foliage, the young foliage, was still showing symptoms of calcium deficiency because there wasn't enough there for the xylem to carry up. And it was in the sap test, as I recall. It was evident. By that time, he had a serious problem. He had 4 acres of tomatoes under a greenhouse in the middle of winter that were starting to flower, and they were all calcium deficient. I'm glad I'm not his consultant, I can tell you that.
Okay, thanks for that explanation.
Okay, so we already talked about manganese. You have these in your notes too, so whatever I didn't cover, you'll get. But here's a chart that I made of the ammonium levels from these test results in the sap and the corresponding levels of sulfur and iron. And this goes back to what I said about the iron appearing to decline, but yet that's what it said in the sap. But the fact that the ammonium was being processed and was low tells another story. Now, of course, the molybdenum might be part of that too. We don't know exactly. But will I see this again? I don't know. I'm going to keep my eyes open for this kind of pattern. Because we know sulfur's not mobile, so it's not going to change much, but iron can be. So I'm keeping my eyes open for this kind of thing. And I made a note here that if we think of it this way, that SAP is a metro, that's the public transportation around Washington, D.C. is called the metro. I think it refreshes my perspective on what we're trying to accomplish with the SAP tests. Let's see what else we have on here. I started at the bottom this time. In the broccoli plants, boron deficiency has been shown to increase nitrate levels in sap. So if we look over here at the boron levels, you see I wouldn't say they're consistent, but they're all between 1 and 4 parts per million across the board. So that's a pretty narrow range. And if you look at the nitrates up here, as I said before, they were going down. They were declining. So that indicates that they were being used and that the plant wasn't getting an excessive level of nitrates. So the take-home message from this is that these boron levels are probably at least adequate. So in other words, if I'm looking at these tests next year after 3 or 4 weeks and I don't see the boron crash, I would expect that nitrate levels will continue to go down. And adding boron is not going to hurt the system. Now, zinc is a rather interesting micronutrient. It's so vital, and as Gary said, it's overapplied. But yet, that doesn't always mean you're going to see it in abundance in a plant, which is ironic, like many things are in agriculture. It's one of those nutrients that's severely affected by phosphorus when that gets too high. And in Pennsylvania, we don't see that, so we don't have that problem. I found this interesting, that zinc is low in phloem, generally speaking. So that says that the zinc that we see in this sap is probably largely being is probably from the xylem sap. And it's interesting that the old sap has the lowest amount of zinc, and the old leaves are sources, which would imply that phloem sap is probably important. I'm not going to say that this is mostly phloem because I don't know, but what I'm saying is an old leaf is a source leaf. So phloem is an important component of that leaf's everyday life, and the zinc is low in those old leaves. Excuse me. I'm going to put myself on mute here for a minute. Incidentally, boron uptake will increase when zinc is low. Now, I don't know if you're going to see that in every test you look at, but that's a trend that's been identified. And so is chlorine. Same thing happens there. So on this test, it kind of looks like that's what's going on if you look at it over the big picture. Chlorine came up in the old sap as zinc was declining to some extent. You know, it's not always exactly what you expect. But the boron was also coming up, increasing. And I have to say that with these tests, you need to have at least 2 sets to start determining if something's changing. 3 is actually best because you have to be able to see patterns. Now, here's something interesting from an insect perspective. Thrips are a very destructive pest that transmit disease and are very persistent. And they are often associated, at least in one of the studies that I've seen, they've been associated with tryptophan levels. And when zinc levels are low, tryptophan levels tend to increase. So does that mean that low zinc could be a warning signal that you could get a thrips outbreak? I don't know. Time will tell. Digging into the literature will tell, but it's something to keep in mind.
One other stimulant for thrips, Nick, found on carrots out in California, high nitrate in the sap. Thrips just thrive to that. They just go nuts over it.
Yeah, that's an issue with a lot of insects, especially sap feeders, like aphids, psyllids, whiteflies, to some extent thrips. Thrips don't actually feed on sap exclusively, but they scratch away at the leaf surface and they get a lot of sap. And high nitrate levels are, relative to other nitrate levels, are usually associated with bugs. So it's something, like Gary said, that's something to keep in mind looking at the tests. But this might interest you potato guys in particular about zinc. As zinc deficiency is incurred, potassium can be exported out of roots. which could have implications for potato management. That is, a zinc deficiency increases export of amino acid, sugars, and potassium out of roots. So actually, when you're...
Hey, Nick.
Yeah.
Nick, that export out of roots, so it's sending those nutrients out of the roots into what? Into the soil or into the other sinks in the plant?
That's a good point. I don't know. That wasn't part of the description I got for that phenomenon, but that's definitely worth investigating. I'm not even going to speculate. I'll throw another one out at you about micronutrient interactions. Zinc, magnesium, and iron are all divalent cations, and they're all similar in size. So if zinc in particular is excessive, it could cause a deficiency in those other two. And that may show up as chlorosis, but it may show up on a SAP test before chlorosis occurs. I'm guessing, and this is, I guess, again, speculation, but it makes sense that if they're all very similar in size and in their valency, that they'd be competing with each other for transporter proteins in the phloem, or in other transporter like channels and enzymes, and that competition could result in those deficiencies. Let me see what else I have going on here. I think there's something about copper yet. Again, since copper is an important ion for several enzymes, overdosing copper, so to speak, is possible, but using prudent rates of copper nutrients is not likely to do it, in my opinion, but I could be wrong. And that reminds me of something else that I didn't mention about zinc, but it's important for copper too. Both copper and zinc are critical for pollination. And I know in the case of zinc, it's important because it actually helps with pollen tube formation. And as I recall, copper is necessary to rupture the pollen tube so that the pollen can actually fertilize the ovule. So now in potatoes, I know they flower, but I don't think that the fruit that they form is economically important, is it?
No, not in the way that we think about marketed fruits. It could be significant in it's another sink.
Right, so that raises a question that sometimes comes up when, depending on what you come across, that I'm not saying copper and zinc deficiencies are good, but if in that case, if you don't want that sink there, it's better if they don't actually get transported to the flowers. But on a tomato, it's obviously there's a point in the phenology when zinc and copper levels are imperative that they be high so that that pollination event occurs properly. And in the case, well, with tomatoes, it's a no-brainer because we have target values. And fortunately for most fruiting vegetables we do, not so much with cucurbits, but as we study the tests and learn about the interactions, and you know the crop phenology, you know, when zucchini plants or squash plants are starting to form flowers, that's a good time to apply the micronutrients, maybe in a standalone form, if the previous sap test or two was showing that there was no risk of toxicity based on what you know about their interactions and based on how the sap concentrations may have changed over a 4-week period, because we sample every 2 weeks. And I'm thinking out loud, but that's the approach I would take, especially on a crop that doesn't have target values. Now, Michelle, I had some things in here about like packaging samples to ship. Do you want me to show that?
Yes, please go ahead, Nick.
Okay. We have about, what, 45 minutes? That won't take 45 minutes, though.
That's just fine if it doesn't. If we end early, I'm not going to be disappointed.
Okay. Oh, here's something. There's something else I want to emphasize here that for this group in particular is going to be valuable. Do you see my screen there? It's got, it says worst data set.
Yeah, we can see it.
Okay, this is pretty important actually from a sampling perspective. We had a client last year who was doing this for the first time. I would say, and this is from one consultant to another, I would say he was not very cooperative, and I'm sure you can identify with that. He bought probably 12 or 15 sets, but he didn't commit more than he didn't commit a good number of them to any one greenhouse. He kind of spread them out. And what happened was early in the season, he sampled young leaves because that's all there was there. And then he didn't come back 2 weeks later or even 4 weeks later to sample that greenhouse again when he had old and young leaves. He came back 2 months later and sampled them again. And you can see why I call this the worst data set. It's worthless. And he didn't sample again the rest of the season on a crop that could easily go 18 weeks. That's not a good use of a client's money. And we can't tell him anything from this. And that's a horrible data set. Now, I'll show you another one that is from an 8. It's from like a 4-week period. Is it 4 weeks or? It's 4 samples of the same. That shouldn't say 4 weeks. It's actually 4 samples of tomatoes. And you see how the resolution improves significantly here. With 4 sets, you can actually see a trend occurring with the old and young potassium sap. You can see that the young sap the old leaves are pumping calcium or potassium into the young tissue, and it's being consumed and cycled through. And then by the end of June, he was in a situation where he was facing a deficiency. Oh, actually, yeah, it's a deficiency because there's more in the young sap. So that young sap was actually had more in it than that plant would be able to sustain over the long term. So a management decision at this point would have prevented this right here.
Nick, we've got some history behind us on plant tissue sampling and primarily in potatoes. And the way we've done it is typically have made available a set of 5 samples we would look at in a season. So the idea of multiple samples during a season won't be something totally new to the farmers that we're working with, at least regarding potatoes. When it comes to something like a small grain or a cereal grain for the spring planted, would we still be looking at well, I guess we would be looking at multiple sampling occasions if the farmer really wanted to go that route. But how would we figure out how many would be useful if it's wheat or barley, for instance?
Yeah, that's a good question. It's something we encountered last year, and we had a consultant who actually sampled spelt and barley. And as I remember, I think he might have been able to sample it 2 or 3 times at most every 2 weeks. So he got about a 6-week window. Would that sound about right?
Yeah, if it's on a 2-week window, I imagine that you can't do much more than that.
Right. And so the direct answer to your question is how do you determine when? In a case like that, it wouldn't hurt to sample every week if you could afford it, and if you could put the input in time, if you could get the results back.
If we're using it as for instance, if we ever figured out how to use this thing as a nitrogen management tool, then most of our inputs for nitrogen on cereal grains in this area, at least, happen within a month or a month and a half after planting, and then after that, it's just growing out the crop.
Yeah.
If I could speak to
So
that,
if we're going to affect how much nitrogen, yeah.
We don't advise growers to sample beyond the point that they are going to be applying inputs. So that's specific to the larger cropping systems with lots of acreage, the grain crops, the soybean growers. We find that because of the large acreage, many of them do not need to invest that much in testing, and then also it's outside of the labor resources to be able to continually sample. And then additionally, if they don't anticipate putting more inputs on as a result of nutrient testing, then that's the point that they're no longer going to be sampling.
That makes sense. But my comment on the nitrogen component, we wouldn't anticipate using much nitrogen in a mid-season application on a grain crop. There could be utility if for some reason we get out of balance and our copper goes wacky or our magnesium's all out of sorts. And we've got irrigated agriculture, so we've got the ability to make an intervention with a fertilization. We've never done that, or I shouldn't say never, but it's very uncommon, and we typically don't track through the season on plant tissue testing for barley or wheat. In listening to the presentation, it sounds like there may be utility at some point if we knew what to look for in trying it out. My comment on when do you start testing, how much testing do you do early, was more on the nitrogen side, not so much on the total balance or total flow of nutrients in the crop through the season. Because we've got the ability. It's just we don't know if we need to or not at this point.
Yeah, the most honest, direct answer I can give you on when to start for the nitrogen management in particular is I don't know.
Okay.
I'm not afraid to admit that, but I also am not afraid to try to find out either.
I might add to that a little bit, Patrick. You know, when you deal with some vegetable crops, spinach is only in the ground for 45 days. So I'd say on your first season through, more for investigatory reasons, you could do sampling each week to find out what actually happens, and then you can sit down over the course of the next planning portion of the next season, determine whether or not you want to go in there and mitigate anything with a foliar application or irrigation through your pivots. I know that's one thing that we are considering heavily is timing of nutrient applications is a major deal when it comes to when they're taken up and when they compete for other nutrients. The potassium, calcium's one issue, and then the nitrogen's another. There's certain times of the season applying certain forms of nitrogen is better than others. So you can take that sample, you know, on maybe one set, an average field or something like that, heavily the first season, and then decide if there's any kind of impact that might be advantageous for split applications.
Now I'll go one.
On the
I'll
side
go
of
one more.
alfalfa, where you've got multiple,
I'm sorry. Go ahead.
where you've got alfalfa, you're going to have multiple harvests per year. I know there was some discussion about alfalfa at the Santa Clara conference. I remember distinctly the difficulty of getting the sample, of collecting the sample that was discussed. Are there any innovations or is there any resolution on how best to sample alfalfa to make it not tedious but also worthwhile?
I did speak directly to Nova Crop about that. The questions I asked them were, do we need to have 2 samplings for each set? Meaning, do we need to sample the young against the old? Is the nutrient flow going to be significantly different such that we need to do a young and old sampling? And they followed up with yes. From their own findings, they still would not only recommend but somewhat require that we sample young and old together to have an actual set. Secondly, if there was a way to reduce the number of leaves, the quantity of leaves that's needed, and they said that because of the testing, or I'm sorry, the processing in-house, that they still need to have that 80 to 100 grams as the absolute minimum. So in terms of saving time, we have not found anything that is a significant time saver other than going through the field and picking the lower level leaves, and you can strip the leaves off of the parent stem instead of having to pick each one. But ultimately, we're still going to have the same quantity that's needed and still going to need a young and an old sample.
Is there a way to gather a large sample of alfalfa stalks, for lack of a better word, the standing alfalfa, and then strip it off at the edge of the field? Instead of walking through picking leaves? Or is there some sort of time component?
Could you specify what you
I'm
mean?
trying to get in my head how we would manage to get through all of our alfalfa field.
Yeah,
How long after you take it out of the field do you need to have the leaves plucked off of it into a bag?
Oh, I understand. Before it starts to show signs of withering. So if you can pick it, and then there's also very, very large Ziploc bags, typically used for clothing storage. But for the larger crops, and especially in the more in the regions with higher temperatures, so say California, they ran into the same thing when they were getting almond leaves and then kiwi leaves. So you can put these leaves, pick them, and put them in these gigantic bags clothing, and that way you can go through the field and still retain moisture. You can also put some, if dry temperatures are a concern, then you can put wet paper towels at the bottom of the bag to retain moisture in the container. But to directly answer your question, Patrick, just so that the leaves don't show signs of withering or dehydration prior to packaging.
Okay.
So this next slide is, I'm not going to, you've seen it before, but this is what I consider the best data set of the three that I showed you. And it's obvious that the resolution is excellent over time. And as I mentioned before, this is not ordinary. This was done by a consultant who's actively, he is doing many trials on his farm to understand the sap testing. But you can, the point of my last three slides is to show you how much better more samples are if they're affordable. And you can get much more information at any stage of the process as you accumulate data sets by doing as many as you can. So one last thing, I'll just go through a few quick. I'm not going to talk a lot about them, but I want to show you some interesting things that as agronomists you might find interesting. Here's a slide that shows how light levels affected sugar percentages. The bars that are in the red rectangles were collected between 4 and 5 p.m. in a high tunnel, and the other bars were collected, those samples were collected in the morning. And I'm drawing a blank on which is old and young here, but as it's obvious that collecting the samples later in the day yields more sugars in the end result, which is not new information, but interesting nonetheless. Here's an example of some nitrogen data. The blue lines are total nitrogen, and the green lines are, I think it's nitrate nitrogen, but I've got this menu blocking my view here. Yeah, nitrate nitrogen. So what I like about this is that you can see here clearly that the total nitrogen levels are robust, but the nitrate levels in the young leaves are rather low. So as the nitrate levels decline, you're still going to see plenty of total nitrogen, which indicates the conversion is occurring. If you look at this graph, this is 7 different tomato growers. If they have red arrows, they're organic. If they're red, they're conventional. If they're blue arrows under them, they are organic growers, pesticide-free guys. And what's remarkable about this data set is that they're from different farms over different time periods, different numbers of sample sets, but there's a remarkable amount of consistency across these 7 sites in terms of how these nutrients were achieving, I call it homeostasis or balance. I use these terms loosely, but that's encouraging to me, because that tells me that the trends that we see will be consistent across farms. So you wouldn't expect a lot of variability between farms in regard to how the nutrients are behaving in the plants. These are all different varieties, different growing systems, nutrients, soils, water, and it's amazing. I thought that was really cool.
Would you be able to classify, if you go back, those examples as examples of good production systems, or not necessarily?
That's a good question, because I don't know who the growers are. I just gave them dummy codes, you know. But the Keystone growers, I couldn't tell you, Patrick. I don't know who's who here, because I can't. I would base that estimation on their outcome, But thinking back on the people that I use for these type of graphs, I tend to pick certain clients from my database, and I'm pretty confident that these represent some growers who were growing a good product that was marketable at a high level. That would be my guess.
That's sometimes a question that comes up as to how a particular lab or consulting company's standards are made. Is it based on just a large population of data having been run for a particular crop, or is it based on a large population of data having been run for an especially successful crop, as to how the standard was set originally? So that's something to just kick around a little bit, and maybe you already know the answer to that. I think that was raised at the Santa Clara conference too. But
well, I know sure at Nova Crop Control, they based their tomato standards on like 10,000 samples, and they do their own trials where they determine what's good versus what's normal versus what's poor. And we try to do that when we do our target value trials here in the States. At least in Lancaster we did, but it's hard because it's difficult to find a poor site on purpose, and everybody wants to be good, so it's often difficult to take the sites that you have available to you and parse them out on the quality of the production, and that's one aspect of determining these standards. So it's actually both. It's what you said is both. It's the volume and the quality. And ideally, we would have the optimal number and a poor site, like a normal site, and then an excessively good site.
Okay.
Now oh, sorry, go ahead. I'm just going to cut to the end here because I know if we could end earlier on a Friday, that's better. So I'll just do something kind of fun. This doesn't require any note-taking or anything of the sort. I'm just going to show you some slides of some of our experience this past season at Keystone. At the I got to get rid of my screen sharing bar. At the height of our season, we were getting stacks of shipping boxes like this from all different places: FedEx, U.S. Postal Service, UPS, you name it. That's what we were getting. And here's some of the other variety of boxes we got. Got shoe boxes, beat up old boxes. People use anything. This is an example of a plastic tub that we had. We had set this tub up at a co-op where the growers would give their samples to a truck driver, and he would put it in a box that we had given to the co-op. So there might have been 5 truck drivers, and they were all giving us samples to ship every 2 weeks. And it made it really nice because those growers, it was very easy for them to submit their samples. This guy was nice enough to send us onion leaves in a styrofoam box with cold packs and a note to send his box back if we could, but we never did. I got a sample in a cracker box once. I'm not making this up. That's how it came to us. We got a sample in a cereal bag. Actually, it had cabbage, worms, and flea beetles. And I told the meeting Wednesday, my favorite one was when they sent us sap samples in the envelope of our competitor who does tissue sampling. There's a box that's about ready to go to FedEx, and a bigger box with cold packs. And at the height of our shipping, we were shipping out 2 boxes at a time, like that. we don't like to see this. Poor quality, not stacked, no identifying information about why they're sending these nasty leaves. And then we also don't like to see them overstuffed. So Michelle will give you standards for how much material to put in the bags, and that's really what they want. They don't like it when you send them too much. That's all I got. So hopefully that was enjoyable.
Thank you, Nick. Michelle, on getting set up so that we can send samples in, we've got a block of samples reserved. We need to do some data input, I think, on the Nova Crop, or is it on your directly through Crop Health, or is it at Nova Crop that we've got to enter data in?
Any communication regarding sampling and ordering would go through me. And then for technical assistance, as we discussed before, because we want your program to succeed with the potatoes, then for technical assistance, we would directly connect you to the laboratory and the consulting team at HortiNova and Nova Crop Control.
I was thinking about the graphing interface.
Yes, the graphing interface, we're going to have a webinar with some additional consultants. It was actually noted by Gary, who presented earlier, that he's looking for a specific sequence of being able to chart graphs over time. And so we're going to have a webinar hosted by Crop Health Labs where the Nova Crop staff directs consultants on how to navigate that interface.
Okay, but when we set up as a client of Crop Health Labs, it'll be through a Crop Health Labs website. Is that right?
It's accessible through the Crop Health Labs website. It's also accessible through the Nova Crop Control website. But if you go onto the Crop Health Labs website, crophealthlabs.com, there is a link on the homepage, Access Database, and you can access it through there. Now, in order to access anything for agroengineering, there would have to be an account that's made. So we would have to first do that. And then with regard to getting labels, so a label is ordered prior to sampling. Reason for that is because it's barcoded, and so once it gets to the lab, that expedites the processing and means that the leaves do not sit idle for any longer period of time than they have to. But at the point that you have clients that are ready to order, we can go ahead and process those. Give us 5 days to get those labels to you, or we can email them to you within 24 hours.
Are you ready for us to set up an account with you?
Yes, at any time we can take that.
Great.
And then speaking again to some of the shipping and sampling logistics that Nick mentioned, we do have specific protocol for each crop. And while there are some similarities across the different crop lines, prior to sampling and prior to shipping, we would want to have a phone conversation at least to go over timeframes for sampling, how shipping and logistics will be worked out, anticipated timeframes for results, which is usually 5 to 7 days, and then how to get your clients best organized so that it's a streamlined system.
Yeah. Okay. Is there, I don't want to sound quite like a broken record, but on the side of what do we do with the data once we have it, most of the people who will be engaging, the farmers who will be engaging on this trial effort of ours, will be keen on knowing, well, what do I do with this stuff? Usually when we do a study on a field, we let the farmer know what we're up to and keep them in the loop on how things are going. We would definitely like to be able to advise if it looks like something's out of whack based on what we've heard on these nutrient interactions and on Nova Crop's guidelines of poor, optimal, or excessive, to have a short list of if we need magnesium, here's the kinds of magnesiums that as a in-season application we might look to, so that we don't have to reinvent the wheel.
Yes, and in terms of guidelines for that, we have input suppliers that. Do you folks have your own input line at Agroengineering?
No, we are solely consulting.
Okay, solely consulting. So yes, we do have established lines of products that have been tried and true that we will recommend. Additionally, if there's others in your area that you want to work on a more regional basis for sourcing, that's fine also, but we do have access to product lines that we recommend. And that would be part of it, basically, because we're establishing you as a new consulting group. Technical service regarding input recommendations would be available to you through Crop Health Labs and even more directly through Nova Crop Control themselves.
Okay, and is this something that we've purchased the sampling if we access Crop Health Labs or Nova Crop, then that's on some additional fee?
No, it comes with it.
Okay.
Now, if there are external clients, say for instance, if your clients come to us directly and want to set up consultation, then there's a $35 charge for that. But for our distributors, no, we consider that as part of the order. It's just considered technical assistance at that point.
Okay. Well, if there are any updates on sampling protocols, if you wouldn't mind sending those on. I think on Nova Crop's website there's, and maybe on Crop Health website now too, there's sampling guidelines for particular crops. So we'll make sure we've read through those and everybody who's going to engage this is on board and they know.
Yes.
So we've got a couple months before we've got anything up and growing big enough to sample.
Great. And Patrick, I set up that Dropbox. As long as you can access that, we'll continue to share documents through that portal. And if you need me to add any more of your team members to that Dropbox, let me know so that you have easy access to it.
I'll just make sure I'll go there now and we'll download them all. See what's there now.
Okay.
So I have 2 files. Are there more than 2 files?
There's also a video feed of the SAP analysis seminar in Santa Clara, a copy of that for your staff.
Oh, okay. Yeah, that was actually a limitation for us because we had the jump drive. You can't. Well, we weren't able to take it off of the jump drive, so.
It was, yes, it's protected. So yeah. I will. I'm going to establish a different Dropbox for you. I'll send you an invitation to that, and it'll just be that way you can share it with whoever on your team.
Yep.
I'll just re-coordinate that.
Okay, that's great.
Okay. And then again, in follow-up, we'll definitely arrange within next week. We'll try to set a time frame, Patrick, that one of our consultants, whether that be Gary through AEA or Nick or another CHL consultant, who can come and sit down with you and your team and do something more in person.
Okay. That would be great.
Okay. Wonderful. Well, I appreciate everyone's time. I know this was an extensive session and there was a lot of information shared, a lot of really positive questions. So I appreciate the questions most of all. I've made note of a lot of them so that we can address them later on. I need to do some footwork on them. But I will keep you posted also on the webinar that's coming up to go over the bemesting online so that everyone can access that data.
Great. Well, Michelle, thanks for coordinating this all, and Gary and Nick, thank you very much for your input. We're very appreciative of it.
Excellent. Everyone have a great weekend, and we'll catch up.
You're welcome. Thank you.